a tribute to Ricardo Galler, and as my co-chairperson is going to announce, Dr. Akira is going to talk a little bit about his career at Fiocruz. He's a very important scientist in Brazil. And I'm very pleased and very honored to be here today sharing this session and seeing that finally this institute is a reality, or this platform. This started many years ago. At that time, I was director of Butantan, and I met Dr. Brechot in Rio in a party offered by the French embassy. And in this party, Dr. Brechot told me the interest that he would have to have a branch, something going on here in Sao Paulo, and he would like to have together Sao Paulo, Rio, and Pasteur Institute. And then we started working on this. The first idea would be to locate this platform at Butantan Institute, but then talking to the rector of USP at that time, Dr. Zago, he convinced me that perhaps it would be better to stay here at University of São Paulo. That's what we did. And finally, there was someone that was always engaged in this, since from the early beginning. This is Paula Minoprio, and she's someone that really has a punch to do things, and we are delighted to have her here because she was the person that could bring this to accomplishment together with Professor Luis Carlos, that also is, always dreamed of having something very international here at University of São Paulo and could help accomplish this. So we're very happy, and today in the evening, the people that have not been there will see this new institute that's very nice, and hopefully we're going to do very good science here. So we're going to start, and so I'm George Calil. I don't know people that know me. I am from University of São Paulo, and also I am the chairman of the coordination committee of this platform. And now Carlos Eduardo Grou is going to present the first speaker.
Good morning, everyone. On behalf of Dr. Rodrigo Correia, Vice President of Research and Biological Collection of Fiocruz, I would like to thank you, all the organizers, and say that it's a great pleasure to be here to introduce Professor Akira Roma, a former director of Biomanguinhos and a former President Oswaldo Cruz. He is responsible for many advances achieved by the production area of Fiocruz. Dr. Akira will start to say a few words in tribute to Dr. Ricardo Galler. Thank you, Dr. Akira. Acho que a gente assiste aqui. Se quiser, então.
Good morning.
Que é melhor para assistir também.
Good morning, Jorge Calil, Carlos Eduardo. Thank you very much for the introduction. And I must also thanks to the organizer in order to have this session in tribute to Ricardo Galler. This is a very important session for us because really Ricardo Galler was a very important researcher, scientist, very strong and deep researcher. He really has studied hard in subject related to molecular biology and in its application in development of viral, parasitic, and microbial diseases. So he really was a very important researcher there and had, of course, unfortunately, a very short professional life, unfortunately, but it was a very intense life. Therefore, it is important to pay a tribute to him. So let me start to have a short presentation where, well, próximo, this side, no? I press this side. Okay, so as I said, he was an important molecular biologist and involved in flavivirus. Flavivirus, and studied the flavivirus genome organization. Here, his picture there, and there he studies genomic organization, expression, replication of flavivirus. Published a paper published in 1990 with Chambers, Hum, and Charles Rice there in the laboratory, and also studied, made molecular analysis of yellow fever vaccine strain, our strain, 17DD strain, with the post Claudia Santos, Ricardo Carvalho, Oscar Souza Lopes. Maybe some of you remember Oscar Souza Lopes was a researcher at the Instituto Adolfo Lutz, also in arbovirus. And we had Oscar Souza Lopes also working at the Biomanguinhos in 1985, something like this. And they published this paper, molecular analysis of yellow fever vaccine strain, in 1991. And he also did a complete nucleotide sequence of 17D strain, 17D2013, which is WHO yellow fever vaccine strain, and our 17DD strain. And they compare these two different strains. There with Carlos Morel and Marcos Freire. And also studied the molecular basis of virus attenuation and its use as expression vector. You know, using this approach, Dr. Lee from NIH developed further and with the dengue vaccine, and Sanofi Pasteur took over this technology and developed the dengue vaccine using yellow fever backbone as backbone, 17DD as backbone there, including there the protein M of serotype 1, 2, 3, and 4, and has reached here now. We proceed with this technology there, but we are still in very initial stage. But, well, this few slides to show the importance, the work developed by Ricardo Galler in this area. But besides flavivirus, he also yellow fever. He worked with yellow fever, dengue, but he also worked with HIV virus, trying to incorporate and use this technology in HIV vaccine development, and also in malaria, pneumococcal bacteria. So he was a molecular biologist looking for application and to come with a product. This was Ricardo Galler. And unfortunately, he had a very intense scientific life, but short. Well, my job here is to talk about the history of yellow fever. And most of, I think I'm not going to say anything new, but just remember yellow fever started there in Africa, and monkeys, and sometime he infected men. No? And we have a first yellow fever epidemic recorded there in Yucatan in 1648. And there was already a suggestion that there exists a mosquito vector, and the mosquito vector and the virus that were introduced there during the slave trade 370 years ago. So the virus was introduced in Americas in 1300s. No? And the yellow fever, the term yellow fever was used first in 1750. An outbreak occurred there in Barbados. In 18th century, yellow fever became a major problem for all colonial settlement in Americas. The origin, in 1793, it occurred several epidemics in Americas. In Philadelphia, at that time, 1793, 10% of population died. 1878, 13,000 deaths in Mississippi Valley and several settlements around this Mississippi Valley. And through 18th, 19th centuries, caused several epidemic deaths in several regions of the United States, in South America, United States. But it also caused epidemics there in Spain, France, England, Italy, West Africa in this time. It was, the virus was spread by the navigation, slave trade, and disseminated, which disseminated the vector and the virus to this region of the world. And Carlos Finlay, he made the discovery of the vector and the yellow fever virus. In 1881, Carlos Finlay presented at the the Cuban Royal Academy of Science the theory of mosquito transmission of yellow fever. And in 1900, the Yellow Fever Commission, headed by Walter Reed, Carroll, Leger, Agramont, showed yellow fever control through Aedes aegypti control, through the vector control. And in 1901, they eradicated yellow fever by controlling Aedes aegypti. And they did the same, the control of Aedes aegypti in Panama Canal construction, and eradicate yellow fever in that region. And in 1901, really, Reed and Carroll did experiments in human volunteer in Cuba. Of course, today it's not possible to do this kind of experiment, but they did experiment on human volunteer demonstrating that the filterable agent transmitted by Aedes aegypti caused the disease, yellow fever. It was the first demonstration of a virus causing diseases. But we also have a publication here showing Brunet, Kozak, Magali. It was in publication of 2009, and they said, they wrote that yellow fever caused 200,000 deaths annually, 200,000 cases annually, and 30,000 deaths in this African region and Latin America region. This endemic region, considered the endemic region. Yellow fever in the Americas, as I said, started 334 years in Brazil, since 1685, as I will show you. So, we have a yellow fever, record of yellow fever, 3,000, this was 300 years ago. There, coming from Africa, there, and showed that, in that epidemic is there, in Yucatan. And then, 1494 in West Africa, Barbados, and it was denominated yellow fever. 1648 in Mexico, Yucatan, yes. And 1649 there in Africa, epidemic, severe epidemic. 1668 in United States, Philadelphia, just to, I already said, 69 also there in Valley, Mississippi Valley. And in 1685 in Recife, there we had a severe epidemic. The virus brought by navigation from the Central America there, and from came down to Recife. And we had a ship from Green Cape via Santo Domingo, and dissemination of epidemic waves with thousands of deaths, and spread out to other regions. You know? In 1849, introduction of in Rio de Janeiro, again through the United States ship, Navarre ship, coming from Bahia with sick people. 1850, we had there only 266,000 inhabitants, and we had 90,000 people sick, and 50,000 people died. No? Severe epidemic. In 1873, we had again a new epidemic there, new wave, and we had 3,700 deaths. In 1876, we had again 3,500 deaths. In 1903, the Rodrigues Alves president appointed Oswaldo Cruz as director of health services, and Oswaldo Cruz adopted the Finlay theory of Aedes aegypti vector transmission, established a whole program to control and eradicate yellow fever from the Rio de Janeiro city. So in 1907, he eradicated it, and no yellow fever case happened there in Rio de Janeiro. In 82 years ago, we started the yellow fever vaccination in Brazil in 1937. But just to let you know also that in 1942, we had the last urban case of yellow fever, urban case in Brazil. So, then, in 1958, we eradicate Aedes aegypti. It was reintroduced, but in 1973, we eradicated it again. So we did it in 2 occasions, eradication of Aedes aegypti from the entire country. But it is important to have the entire region without the vector. At that time, we eradicated, but some countries did not. So Venezuela did not eradicate. And the United States was not in favor of eradication. And then, again, the vector came back to Brazil. And today, it is spread to entire region. No? And we say, well, yesterday we heard here vector control. Looks like we are very, very far away from the possibility to eradicate. We need to have a new technology, new approach, new way, new vision, new technologies, I think, to come with eradication. Is it possible? I think it is possible. We must proceed with this, you know. Then, just to show you, I have data here since 1930 in Brazil, yellow fever cases and the yellow fever deaths. So we had always very high deaths, incidence, very high incidence there, 1930s. But then came the vaccine and came down the number of cases and deaths, but came back again in 2017. You can see here a very high case, number of cases of yellow fever and death. Just to show you the evolution of yellow fever in risk areas, you can see here 1997 through 2008, the expansion of endemic areas in green, and then you have red areas which was transition areas, were considered transition areas, and endemic Indian, no Indian region in blue. But the last before the last, the third map there, you can see the yellow stop the expansion area, and the last one, you can see that all the transition area was incorporated, and the region recommended for immunization expanded a lot there. And here, in January 2019, you can see that almost entire Brazil is recommended for vaccination. Today, the Ministry of Health recommend, the National Immunization Program recommend to have immunization in the entire country, you know. And you can see here that we had severe episodes with 1,000 cases notified and 48 cases confirmed with the virus on non-human primates. Here in the southwest region, São Paulo, and coming to, and yes, this one, you can show in the right map the coverage, which is very important to let you know. You can see that there were many regions with poor vaccination coverage, even in the region recommended for vaccination. You can see that, many regions with less than 50% of coverage. So, and only few with the blue, green, with 80% to 100% coverage. So we had many regions with very bad coverage of vaccination. And just to let you know, we have a yellow fever in the period of always in the season, raining season period. It comes in November through May, June. We have the peak of yellow fever in the countries, in the Brazil. This is a map after collection of data between 1999 to 2011. In Brazil, the recent case of yellow fever, we had between July 16 to February 17, 509 cases and 159 deaths. Look, it is, and in July 17, 2017, up to 30 June 18, 518 cases notified, 1,376 cases confirmed, and 483 cases of death cases. It is terrible for a country like Brazil, which we have vaccine available, and the majority of people here, these cases and deaths, were not vaccinated. It's a terrible problem of vaccination. And here, is 2019, in São Paulo, we have Paraná, Santa Catarina, also is coming down to the south. So this is a new situation we have today of yellow fever. The very difficult situation where we have a vertical transmission vector, no? And the Sylvester area coming very near to the urban area, no? And this contact is very frequent. So, and just to show you the Amazon region, we had there the cycle 1989, and then came down to central Brazil, 1999 to 2010, and 2016 is coming, and coming still. The cycle is coming down. No? I think my time is very short, 2 minutes. Just to let you know that development of yellow fever vaccine was one of most important, I think, the breakthrough of technology for vaccine development. They used the monkeys, used the mice, used the chicken embryo, tissues, and embryo. No? And we received here this the strain 17DD in 1973 by Hugo Smith, and we had also here the development of all stages of vaccine production was done here in our laboratory in Rio de Janeiro. The scale-up, formulation, stabilization, presentation, the production development was done there. In 1 year, 6,000 people was vaccinated, And I can tell you from 1937 to 86, 150 million people was vaccinated. And 87 to 2018, we supplied almost 700 million doses to National Immunization Program. This current year, we are going to supply 40 million doses. And we have today a CMO with LIBS laboratory, a São Paulo State pharmaceutical. And we export also vaccine. We have, unfortunately, some severe adverse events, but I think Tatiana will talk about. So I will go ahead and say that the yellow fever is still a big problem in the world. And WHO has established a program to eliminate the yellow fever epidemic by 2026. I think is very optimistic, because if you don't think also to eliminate vector, only vaccination is very, very, very difficult. So you must combine different actions to have elimination of yellow fever epidemics. Because, you know, while we have yellow fever, Sylvester yellow fever in the jungle coming with the monkeys and so on, the virus will survive. And vaccination, the entire world, I think it will be a big problem. So we need to control vector, at least urban. I am not talking Sylvester vector. Maybe sometime we can think about in the future. But today, urban vector, and then vaccination. I think then we can do this job. The last slide I have, I would say that in memoriam of our colleague, Reinaldo Martins Menezes, a physician, a pediatrician, a very important researcher. Also, he contributed with clinical studies, pharmacovigilance, and yellow fever vaccines, did dose response study, duration of immune dose response, and Tatiana also will talk about. Thank you very much.
Thank you, Dr. Akira. Thank you very much for the enormous contribution to production of yellow fever vaccination. We do have a very good vaccination, and Dr. Akira is involved in this for many, many years. Thank you. I'm not going to open for questions because they're running late already, and then it's my pleasure to introduce you, unless there is a burning question. Do you have, or you can always talk to Dr. Akira. So it's my pleasure to introduce you Professor Esper Calas. Professor Esper Calas, I was very pleased to invite him to come to the School of Medicine, University of São Paulo, to the Division of Clinical Immunology and Allergy 12 years ago. And recently he was nominated as a full professor now in infectious diseases. And he has very interesting observations on the outbreak that we had in Brazil of yellow fever. Esper?
Thank you. Well, thank you all. Thank you for this invitation. It's a pleasure to be here today and to celebrate the opening of this new institute. So it brings a lot of pride to our institution, to the city of São Paulo, and the University of São Paulo. And I'm very honored to present some of our recent data on yellow fever and discuss some aspects that we can advance on understanding the pathogenesis of the disease. So as you know, as Dr. Akira brilliantly pointed out, this is a very nasty disease caused by RNA virus that's well known to everybody. And it's been, for many, many years, part of our history. In fact, I finished my ID training in the early 1993. And back then, it was just, we never saw a yellow fever case. We knew the stories about the early 20th century, vector control, disease control in Brazil, and there were scarce reports coming from north of the country where some people that were not protected with the vaccine would acquire disease, and most of them, about 50%, dying of the disease. Well, it was in 2004 that I saw my first case, and that was when a friend of mine, we used to care for patients in our clinic together, and he had a mission in the Amazon basin, but he was only vaccinated in 1965 when he was in medical school. And he was bitten by the mosquito and got the disease and almost died of it. So for many, many years here in São Paulo, I was the only physician with experienced yellow fever with one case. So, but unfortunately, he survived through the disease, and everything came back to the status that no cases would come up until the end of 2016. And as pointed out also in the previous presentation, Brazil started noticing a surge in cases, mostly in northeast of Minas Gerais state. Here's the state. I was born in this state. And although it was an area where one expected everybody to be protected against yellow fever with the vaccine, this area was seeing and documenting cases that occurred after a lot of primate death was detected by the population. And with that, we start getting concerned about a new outbreak, a sylvatic occurrence of yellow fever. In fact, some of the reports have shown that monkeys from the forest in Brazil were starting to get infected by the virus and dying of the disease. The colleagues working at the surveillance of those monkey deaths warned us that potentially the following year, next yellow fever season, this transmission would approach around the São Paulo area. And they were right. I mean, what happened in the following season was the approach of yellow fever around São Paulo area, and we start seeing lots of cases. A total of 537 cases were confirmed, 183 deaths related to the disease, and the state of São Paulo reported a projected 36.9% mortality. With that, we acknowledged that a sylvatic outbreak was going on, is surrounding the city of São Paulo, and this year, the following season, it went to the south of the state, towards the state of Paraná. With that, we start seeing cases. So yellow fever is characterized by, although many people are asymptomatic, is believed to be asymptomatic, incubation period of 3 to 6 days, with pronounced viremia and a very severe clinical presentation, beginning with a non-specific symptoms, with chills, fever, headache. One thing that's pretty common in patients with yellow fever is back pain, which is a little bit different from other acute febrile illnesses, myalgia, nausea, and vomiting. They had the Faget signal, some of them with low blood heart rate and high fever, and many of them evolve into severe forms of the disease. So we also documented leukopenia, clotting disorders, increased in liver enzymes. That's pretty remarkable. I'll show you more data. And that's a result of hepatocyte apoptosis. Liver failure encephalopathy is one consequence very commonly seen in these patients. And after the end of the first week, beginning of the second week is when we see these severe forms of clinical presentation. And late in the disease, they present with jaundice. This is something that we've been discussing, although the name is yellow fever. The yellow name comes from the convalescent phase of the disease, not the acute phase of the disease, in kidney involvement with nephrotic syndrome. At this phase, about 40% of these patients died, despite a very thorough, inaccessible intensive care treatment. Secondary infections are common. They recover with time. Unfortunately, many of these patients, when they recover, they don't exhibit signals of sequelae. And once you infected, you are protected for life. So with those cases occurring, we had the opportunity to put together a cohort of yellow fever cases. Starting in the first semester of 2018, the suspected cases of yellow fever from our hospital at the University of São Paulo and the neighbor institution caring for infectious diseases patients, the Infectology Institute, Emílio Ribas, put together an effort to get into this cohort confirmed cases based on PCR and based on autopsy. And we established a sample repository and start following these individuals. The team was led in USP by Royer from the intensive care unit of the infectious diseases department, and Carlos Moreira and Luiz Zanella at the Emílio Ribas Infectology Institute. So we included from January 11 and May 10, 2018, 76 confirmed cases. They were distributed in these two institutions. The University of São Paulo was the referral hospital for the more severe cases, whereas Emílio Ribas had the cases that were not as severe. 74 were confirmed by PCR, and 2 we were able to confirm only in autopsy. And as you can see, despite the intensive care they received, 35.5% of these patients died. So the first thing we analyzed was the viral distribution. And if you can see on the right-hand side, I apologize, they're too small, but these are the cases that we had full genome sequence of yellow fever virus, and we were able to cluster them into one monophyletic South America type 1, subtype E distribution. So it's a single strain of virus causing this outbreak. And then we decided to look more carefully in the clinical and demographic characteristics of these patients. So overall, in these 76 patients, we went through the symptoms. The most common was fever, 91%, and headache and myalgia. And then we broke down into groups, the survivors and those who died of the disease, in trying to identify some clinical signals or symptoms that could split these two groups in order to help the clinician decide, make the decision who are the more severe cases. As you can see, there are only 2 things that popped out as clinically significantly associated with death. The first one is age. So you compare here, it's the median age was 40 in the survivors and 50 in the deceased patients, and a concentration of males. But as you can see, most of the cases who acquired the disease were males, so 89% of them. And why is that? This is the characteristic of a sylvatic yellow fever outbreak. When the yellow fever goes urban, usually you have a 50% breakdown in gender. But in this case, those people who go into the forest, go outside home, are mostly males. That's why they acquired the disease. So then we consider gender as something that was related to the disease progression. However, when we looked more carefully in the laboratory findings, then we found several aspects related to poor outcome. So you can see here the most that in the CBC, we saw that increased neutrophils was associated with poor prognosis. And that came to us as a surprise because although there were few reports in the literature, we thought that the problem in fighting this virus was concentrated in lymphocytes. Nevertheless, lymphocyte counts were not different. And also another thing that struck us was the lack of difference in platelet numbers comparing those who died versus those who survived. So you see here 79,000 cells per microliter compared to 67 is absolutely non-significant. Then we moved to the liver enzymes, and as expected, increased the liver enzyme levels were concentrated in those who died of the disease, with AST being the most important. This is another clinical characteristic of these patients. AST is elevated above the ALT elevation, and this helps us to differentiate acute hepatitis from yellow fever and other viral pathogens. And then we looked at clotting disorders. So you can see here there is an increase in clotting disorder in those who pass away, and then increased the liver bilirubins, also elevated in those with a poor prognosis, creatinine elevation, and for the first time, we were able to demonstrate that high virus load was associated with poor outcome. Well, this comparison, as you can see, is performed using univariate models of analysis. So we went ahead and started looking at each of the most significant of them using survival curves. So this is the breakdown. We broke each of these variables based on ROC curves. So we decided what is the best breaking point, and starting with age, it was 45 years old. Those above 45 years old, they die faster. Then we looked at neutrophils, was remarkably associated with a poor prognosis. The breakdown point here was 4,000 neutrophils in the CBC. And the AST was one of the most remarkable ones. Those above 3,500 units per ml had would die early in the course of the disease. Then indirect bilirubin carried this same kind of pattern, and creatinine was also associated with poor prognosis in the survival curve. However, this is something that, although it was intuitive, we're able to clearly demonstrate that virus load was associated with poor outcome. Then we went ahead with the multivariate Cox proportional regression model to look at all these variables that were identified as being independent and it was not associated with another one. Let me give you one example. For AST, we know AST and ALT, they are both predictive of poor outcome, but they are collinear. When we looked at the collinearity, we see that AST carries the effect more than the AST. So then we've chosen AST. So that's why we selected those 6 variables to look in the multivariate analysis. And we found that 4 of them were independently associated with the disease progression. The first is age, the second is neutrophil counts, the third is AST levels, and then last one is virus load. And I'll try later to explore a little bit more why we think these 4 variables were independent from one another. So the effect of age for every year the patient had, it was an increment in 5% of death. For neutrophils, each 1,000 cells increased 21% of death. For AST levels, for each 1,000 units per liter, there was a 10% increase in mortality. And finally, for each log of virus load increase, we had a 27% in mortality. We designed some algorithms using our cohort. Due to the number of cases, we could not do many of variables or many algorithms because the final group's breakdown were small, but the best predictor of mortality is when we combined neutrophil counts and virus load. For those individuals with high neutrophils and high virus load, and the limits are 4,000 here and 8 logs, if they're both positive, 100% of these patients died. If the neutrophils were lower than 5,000 and lower than 8 log, only 11% of them died. So with these exercises, we were able to perhaps start trying to find what are the predictors when these individuals show up in the emergency room with yellow fever diagnosis. And this is, to our point of view, important to guide the clinician. What is the patient that's more important to be privileged in going to the ICU, receive intensive care, and those who can be in normal wards and requiring less attention. So for these four variables, we believe that the first age is a reflection of senescence of the host. So senescence can probably result in a dysfunctional immune system, and that, as seen in other diseases, can be related to poorer outcome. The second one is neutrophil counts, because here, this is quite interesting, because neutrophils are not necessarily directly related to a viral infection. Nevertheless, it can reflect inflammation, it can reflect bacterial translocation, or even the cytokine storm that some of these individuals may experience. So we believe that neutrophils can be elevated due to an indirect effect, but also it's not impossible that could be a result of a sepsis that these patients present in the emergency room. Finally, AST is a proxy for liver damage, direct effect of the virus that comes along with virus load. I mean, the more virus load, more liver damage, more poor consequence for a given case. In that virus load effect is very important. I think it's the most precious observation of this cohort, because it opens the discussion that actually revives the discussion that an antiviral drug can be beneficial to yellow fever cases. Unfortunately, we don't have an available antiviral approved for clinical use. There are some chemical components that are under investigation, but I will call your attention to two drugs here. The first is sofosbuvir. It's been shown by 2 independent groups here in Brazil. They have an antiviral effect in vitro against yellow fever, and it's a drug approved to use for treatment of hepatitis C. With that kind of information, a group in our university and the Infectious Diseases Institute at Emílio Ribas decided to carry on a clinical trial that's underway to investigate whether sofosbuvir can prevent poor outcome in yellow fever patients. So far, 62 patients have been enrolled into this trial, and it's going through interim analysis, and we're going to learn quite soon what is their findings. Everybody's excited about this, and see whether the use of sofosbuvir that was given for 7 days immediately after diagnosing yellow fever can prevent poor outcome. The second one is another drug, is an adenosine inhibitor called galidesivir. It's been developed by a company named BioCrest. It's under study for filoviruses in Africa, but also shown to have in vitro and in vivo effect, antiviral effect for flaviviruses. They have data for Zika virus and yellow fever virus in animal models, and they already went through phase 1 clinical trials to investigate its effect against Marburg virus. So they approached us. We had a protocol approved and ready to go to understand whether this drug administered IV for 12 days can prevent poor outcome. And finally, something that I truly believe is going to replace antivirals in the future are monoclonal antibodies with neutralizing capacity. I think we need to devote more effort in understanding how can we better discover these neutralizing antibodies to treat not only yellow fever, but several other infectious diseases and replace in the future antiviral drugs. So to conclude, yellow fever is a very severe disease. We're able to identify the predictors of death using this cohort. There's no available treatment. Novel therapeutic approaches are warranted and to help treating these patients. I'd like to thank the team that worked in this effort. This is on the left, the team at the University of São Paulo, especially Ye Ho from the ICU, Alice Song, who helped put together the cohort, and Vivian Velino Silva, who did the statistical analysis of the data set, the colleagues from the Instituto de Medicina Tropical at the University of São Paulo, who did the virology studies, and finally the colleagues at the Emílio Ribas Institute that helped put together this cohort. Well, thank you, and I'll be happy to answer any questions, or now or in the end.
So hopefully we'll still have some time for questions at the end, but if there's any question now, yes. Do we have a microphone? Where's the microphone? Okay, speak loudly, please.
Thank you very much for this presentation. Very great. Then the death is much more related to the physiopathology of the patient and not at all to the virulence of the virus. Did you compare sequences of the virus from dead people and non-dead people?
Yeah, it's a great question, because one of the things we thought could interfere with outcome would be yellow fever virus variability or diversity. And we did full genome sequence in 12 of them, and they were absolutely the same, just minor point mutations. And we saw the same kind of pattern for like dengue virus. We did this during the 2014 outbreak, exactly the same. They have very low diversity, so we don't have critical numbers to compare. It would be, we could compare potentially comparing types of the virus, genotypes, different genotypes, but here there was just one circulating. So unfortunately, we were not able to assess this variable.
So have you looked at the cytokine concentrations and distributions in these patients? And if you've looked at polymorphisms, specifically associated to lambda interferon, or?
Sure. So great point. So we are currently doing that. So for the cytokines, we don't have the consolidated data, but when you look at the cytokine distribution, it's remarkable. I mean, the cytokine storm, these individuals that are dying of the disease, is complete nightmare. I mean, it's just cytokines all over the place. whereas those who are surviving have better profiles. But we need to consolidate that data to understand how, what are the main cytokines that are the culprits of this poor outcome.
So your data makes sense with inflammatory and non-inflammatory?
Yes.
It is IL-6 versus IL-12?
Yeah, IL-6 is one of them that popped out as significantly increased in these patients. The problem with cytokines is there are several platforms to look at them, and they seem to vary quite a lot. So we are trying to compare them and see what is the best way to depict the data. So we still have some computer work to finalize. Regarding the polymorphisms, so what we're doing is transcriptomics in those samples. Well, our approach is we got 26 people who died and 26 people who survived, and we did transcriptomes in them, and we are at the moment to analyze the data. Hopefully we're going to have a result quite soon, and our idea is to do a system biology approach to identify what are the cascades and what are the genes involved in a poorer outcome.
I'd like to add that we are also looking for some candidate genes, some polymorphisms that could be involved in innate and acquired immunity. Yeah. That's also being done together.
Okay, so it's a great talk. It's maybe on the top of that, adding that we know that sometimes for the adverse effects for yellow fever vaccine are associated with type 1 interferon mutations mostly. They're rare, but I wouldn't expect those to happen in this population, but I would think that maybe polymorphisms associated with lower levels of maybe in receptors for type 1 interferon or in the pathway could also be one of the genes that I would search.
Sure.
This is actually number one suspect, the interferon and its receptors. So that's what we are studying.
Yeah.
Right
Yeah, type 3 too, but I would
Sure.
I think type 1 would be best.
I think there's
sure.
another one.
Still one more question? Quick question, please.
Okay. How many days after the onset of disease do you see the differences in the profile counts and how many days after the onset of disease? Onset of the disease. How many days after? Because we don't see that.
Sure, it's in the first tape. I didn't make the emphasis on that. But the median onset of symptoms when they admitted to this cohort was 7. And comparing those who survived and those who died, it's the same. So for that, the cohort is well controlled. Because I think it's a great point, because when we see these differences, you can argue that those who were admitted early had high virus load, or those who came late, they were selected biased toward more severe cases. But in that cohort, there was no difference. Now, virus load dynamics is not known for yellow fever. So we understand they have a high viremia in the beginning after onset of symptoms, and they go down with the days passing along, but the exact dynamic is unknown. So this is important to say, because we need more studies to describe that better. And why I'm saying this? An antiviral drug can be useful, especially in those early stages of the disease. If you receive someone who is already in liver failure 10 days after onset of symptoms with multiple organ failure, potentially the antiviral will not serve as a beneficial effect for this patient. Whereas if you see someone coming with not so severe disease, but very early in the course of the disease, where the virus load is very high, so the antiviral effect may be much more beneficial. So that needs to be appreciated when we study this further, and if eventually we start using an antiviral drug, in my opinion.
Thank you, Esper. So let's continue with a very important issue, and I invite Dr. Stella Bevilacqua from the Institute of Biomedical Science at the University of São Paulo to talk about the fetal-maternal transmission of yellow fever virus.
It's okay. Okay, thank you very much for this invitation. It's a pleasure to be here, participate in this such outstanding meeting. Thank you very much for the organizers, and I congratulate also the organizers for this meeting. It's fantastic, really fantastic. I would like also to apologize because I was bothering you with my cough. I'm so sorry. But I will try to speak with my falling voice in this time. I hope it's okay. Okay, I'd like to show you some preliminary results about yellow fever immunization during pregnancy and in a mouse model in which we are studying gestational performance and viral antigen distribution in placenta and the fetus. And I don't need to explain a little much about the yellow fever sorry. About yellow fever vaccine, but it's nice to say that it's a live attenuated virus and grow in chicken embryos. May I have some? Thank you. No, I have some here, but it's better to have more. Thank you. But it's very nice to say that it's a highly recommended the vaccination is highly recommended for people living in or for people traveling to areas, endemic areas, and we have a high potential for yellow fever virus exposure. However, vaccination is not recommended to pregnancy, during the pregnancy, and unless it is inevitable, in general, we don't have vaccination. It is because it's based on the potential risk of viral transmission through the placenta, of course, to the fetus, and associated with the possibility of neuroinvasion and of the virus into the fetal organism. I'm really sorry. However, no vaccination during the pregnancy time also increased the possibility of the disease in the mother and also in the fetus, and compromising mother and fetus survive. So I think it's very need to improve, to increase the knowledge of what happens during the gestation, so that we can really organize better vaccination in this very specific phase. The literature is a little controversial, and we have a lot of papers showing relative risk for spontaneous abortion, congenital yellow fever, and malformation. At the same time, we have another amount or a huge amount of literature showing that abortion, stillbirth, and malformation rates after vaccination is similar to that found in the general population. So it's really very difficult to know some fundamentals from the literature. And in this context, we want to study the effect of vaccination against yellow fever in mice, studying the gestational performance, correlating these data with the placental structure, immunolocalization of the virus antigen, and the viral activity at the maternal-fetal interface, maternal liver, and fetal organisms. Okay. The study was divided in different phases, and here I show some primary and preliminary results from the phase 1 and 2, because the 3 and 4 are still in progress. And the phase 1, we aim to identify potential pregnancy phases during placentation and organogenesis in which vaccination might induce relevant changes. We use mice, pregnant mice, CD1 mice, on gestation days 0.5, 5.5, 11.5, and 13.5. The vaccine was yellow fever 70DD vaccine, and gently provided by Fundação Oswaldo Cruz. And it was the route of administration was subcutaneous at the doses that was diluted to be equivalent to the vaccination in human. And of course, we have a control, a sham control group, and the animals were killed on the day 18.5 for measures like number of placentas, early number of placentas, early and late resorptions, dead and alive fetus, implantation sites, and a lot of gestational profile to organize the gestational profile after vaccination. Why we choose this step? Well, at this point, we choose the 0.5 gestation day because it's the day immediately after the fertilization stage. Also, the gestation day 5.5 is a hallmark in embryology and the embryo development because it's the step in which the embryo is absolutely embedded into the endometrial stroma. So it's the first contact with the embryo with the maternal environment. The days 11.5 and 13.5 are phases in which the placenta is completely differentiated. They are morphologically, functionally working very well. So they have all functional endocrine functions, immune functions, and our regulatory functions are mature. And also we have the development of the organs on the embryo. Almost all the organs are beginning to work and are completely differentiated. So that is why we have chosen these phases, and so we can see the effect of the immunization on these days. As you can see here on this table, the only phase in which you don't have significant differences and the parameters and gestational and the pregnancy parameters is is when the mothers were vaccinated on day 5.5 of gestation. And on this day, you have an increase in number of stillbirths, fetal loss, and of course, a decrease in the alive fats from an average in the liters. So when we analyze also the placental and fetal weights when we administrate the vaccine, also we found significant differences only when vaccination was administered on the 5.5. We have a restriction of the development, a growth restriction on the fetals, and a little bit increase on the size of the placenta. On the remainder stages and remaining vaccination time points, we don't see any difference in comparison with the controls. So it suggests, and we have a strong suggestion, we have repeat this stage, these vaccinations. And they suggest that there is a period of susceptibility to the vaccinal yellow fever virus during the pregnancy. And perhaps this susceptibility is a very short time during embryo implantation. We did some morphological analysis of the embryos of the puppies, and we could see puppies, alive puppies with a normal and apparently normal morphology, and we have also dead fetus. And this fetus was microscopically divided, and stillbirths is absolutely normal, morphologically they are apparently normal, and size and so, but they have no hit, no heartbeat. So probably they are dead, but we consider this as a late fetal death. Also, we have some early fetal death that was characterized by an apparent development delay and degenerative signals, as you can see on the figure B, on the panel B, and post-implantation resorptions that is characterized by masses of non-recognizable fetal extractors as indicated in the C part of the figure. So we go on with the experiments, and in the second phase of the experiments, we vaccinate the mothers only on the day 5.5. And just using for comparison and for analysis is stillborn pups and alive and apparently normal fetus. We use immunolocalization and PCR to identify places and antigen and the viral antigen distribution in the maternal liver and placentas and in the fetal organisms. The immunolocalization of the yellow fever antigen and the sections, I don't know if you are seeing very well, but it's beautiful figures. You can see that in liver of the mother and alive fetus, we have just very few radar cells, reactive cells to the virus. And in contrast, in stillbirths, we have much more reactive cells in different organs. For instance, we have a liver, in ganglia cells, in nerve cells, and brain. So when we did some quantitative analysis, I think it's a little bit more clear that we have much more, it is a prevalence of reactivity than stillbirths, but you don't have in comparison with the alive fetus. So immunolocalization and placenta, on the other hand, also show very similar results in alive fetus. We found some cells, reactive cells, but it's not very evident in quantity. But in the stillbirths, we have much more reactive cells in different parts of the placenta, in different zones of the placenta, in the endocrine areas, and also in the maternal and molecular exchange areas, also in the labyrinthine zones. So also in the stillbirths, we have this prevalence of reactivity, which means that we could find the virus. I think the first result we have to discuss here is the possibility that this virus has crossed the placenta during the fetus was alive or after the death of these puppies. Viral RNA of the capsid protein also was localized, and how you can see on the figure, we have a very low and only traces of viral RNA in the alive fetus, but we have, on contrary, we have much more evident levels in the fetus that has been death-born. It was, of course, confirmed by sequencing of this material. So viral RNA is detectable in stillbirths in comparison with alive fetus. We have several possibilities, several summary points that I think is important, but two of them, it's much more important in my point of view, I hope you agree, is the changes in the gestation performance. It increases birth rates. It's not only that, but losses and embryo losses in general was seen only when immunization was occurred on or happened in the post-implantation phase. In fact, on the day 5.5, in stillborn pups, yellow fever virus clearly reached the fetal organism, fetal organisms, and immunoreactivity to the virus It was predominant in the nervous tissues. So cells and tissues, it's correlated with some data about neurotropism of the virus and the infection. And placental tissues also, and the viral RNA was also found in the fetal tissues. And born live fetus only scarce yellow fever immune positivity cells and traces of virus of the viral RNA were found in the fetal tissues, and liters of the same. It's important to mention that the liters of the same female could contain live and dead and stillbirths and the same litter. And we always found very low traces, very low levels of yellow fever particles in the maternal organism. Well, to take it home, is immunization during pregnancy show it to be safe, or at least is in the direction of to show us that it's safe. The stage of implantation seems to represent a window of susceptibility in which vaccination can, and associated responses may interfere in the course of gestation, and trophoblast and placenta maturity in the latest gestational stages may be the key for avoiding congenital passage for the yellow fever virus to the fetal organisms and gestational changes inducing fetal losses. Well, I think this is just the tip of the iceberg. We have much more to do, but we cannot stop here. But the experiments are not complete. We cannot stop to think about what is happening and what is happening when we have a vaccination during the implantation site. It's a very short period of time during the development of the fetus. So, and if a trophoblast may have a critical role in these tubers after vaccination. So we have to remember that the embryonic barrier during this stage, it's a very thin layer of trophoblast giant cells surround the embryonic area, and the cells are not committed with other functions than to include, to implant the embryo into the stroma and the endometrium. So the cells are quite, has a very high activity and invasion of the vessels surround the embryo. So they open uterine capillaries surround the embryo and establish a direct contact with the maternal blood. In this term, it's possible that the particles, viral particles of the vaccine can reach the maternal circulation, and in this point can interact directly with the trophoblast giant cells. The question is if the immaturity of the giant trophoblast cells at this point may allow the colonization of the virus and placental and embryonic stem cells. At this point, all the embryonic cells are very undifferentiated, as well as the trophoblast that in the later phases will form the placenta. And if this modification, this step or colonization, and this step of the gestation can induce changes that may compromise the fetal organisms in the later stages of the gestation. On the other hand, we cannot avoid the possibility that there is nothing to do with the placenta, nothing to do with the trophoblast. That is a pity because I work with trophoblast. I'd like to see some reactions in that, but it's possible that the immune response to the virus is responsible for that. Well, as have been told in the last presentation, the virus per se, it's able to induce a cascade of cytokines in different times after the infection. It's possible that the vaccine can do the same. And the initial vaccination make it that the exposure of this embryo during all this station to this oscillation in cytokines, that we have increased cytokines, decreased cytokines, different kinds of cytokines, inflammatory, some of them are regulatory cytokines and could be very harmful to the embryo along the gestation. So, as I told you, I could stay here and make a lot of speculations, but it's not the point of this presentation. So, I hope you have a model. We are trying to go on with experiments in order to have a model to study different steps of the vaccination, after vaccination, and embryo and maternal, fetal and maternal organisms and in placenta. And I hope next time we can have a very detailed program showing what happens after this vaccination and gestation. It's a very important point and cannot be left around. So, just some ongoing experiments that include localization in very short times, in very short periods of time during gestation, cytokine kinetics, and viremia estimation in the mother and also in the embryo, and mainly in the trophoblast, and in vitro assays, of course, to use post-implantation trophoblast and challenge with the virus and the vaccinal virus. That is our point just now. So, because science is not I, it's we, and I am sure I have to thank my collaborators, mainly the students that are involved in this process, Fernanda Magaldi and Fernanda Carini, and Dr. Helena Sati, that is the immunization program of the health of the state of São Paulo, and of course, all the others members of our lab from here and out, and the financial agents that is quite important for experiments. Thank you very much again, and have a very good meeting.
Still a very interesting observation. The point is how we're going to translate this to humans.
Yes.
And I mean, live attenuated virus has been a challenge for all regulatory issues, not only here in this country, but for the entire world. And there are many people that say you cannot use attenuated virus for this, and that. And we had made, for instance, some observations, not in pregnant, but in immunosuppressed or immune-depressed people. I mean, even though we have said to people that had common variable immune deficiencies not to be vaccinated against yellow fever, we had 30 that were vaccinated because they listened more to the neighbor than the physician, but they didn't have any problem, I must say.
Yes,
okay.
Thank you.
Any questions? Talk loudly, because I don't know where the microphone is.
Do you intend to better define the area of the increased risk for fetal death?
I'm sorry,
Do you intend to better define the area of the increased risk for fetal death?
Yes. At the next step, we are doing some experiments with different time points, and we intend at this first moment to analyze the post-implantation time points, much more time points after implantation. And the day 6.5 and 7.5, 8.5 and 9.5, but not also to analyze the embryos as this one, but also to look for cytokines, viremia, and the maternal, and to do a correlation between such kind of points.
Okay, any other questions for the three speakers of this morning? No? So we are going to break now, and we'll be back exactly at 11. Thank you. So the next speaker is Dr. Lúcio Holanda de Freitas Jr., also from the Institute for Biomedical Research. Dr. Lúcio that studied biology here in Brazil, and then went to Pasteur Institute in Paris for 5 years. And then for 8 years, he was in the Institut Pasteur of Korea, where have been studying and developed new drugs for many of the neglected diseases. And that's what he's going to tell us about, how he uses his platform to develop new drugs for those neglected diseases. Lúcio, thank you.
Thank you, Professor. So it's a pleasure to be here. So I'd like to thank the organizers, Paula Minopri and Luis Ferreira for the opportunity. I'm very glad to be here. I was in Paris for 5 years, so it was a very great time. I was in the Institut Pasteur in Paris in the time that most of the trainee people, they are from Brazil. You have so many Brazilians there, it was such a good environment at that time. You have Professor Edobrando Pereira da Silva, Professor Michel Rabinovich, Paula Minoprio, Denise Matei. So it's a great time. So I really wish for Pasteur Brazil great years ahead. So I'm going to talk today for you guys about the work that you have done doing this screening for yellow fever virus. So our lab is pretty specialized. Drug discovery is something very complex and expensive, and our group become specialized to develop phenotypic screenings. So in our lab, we have something like 22 different assays established for Chagas disease, worms, dengue, different tropical virus. And this is what you do. So, and you do this job in the Instituto de Ciências Biomédicas here in USP. In this lab that I'm working here in Brazil, in USP, was a lab that was completely renewed by a pharmaceutical company, Europharma. So this was very important for us. It's important to do this important work, but you have to be connected with the pharma. The question is to be sustainable. You cannot depend only on public funding. You should somehow to bring the pharma to do projects in partnership. So this laboratory was completely renewed by Europharma. This is what you do in our lab. So what you do, you're trying to mimetize what's happened in the human host when the infections take place. So in this case, you have then, this is the establishment of the dengue assay for high-throughput screening. You have here, so what you see in blue is the human cells, and what you see here in green is the dengue virus. So what the people is doing here, so they're trying to find the best conditions, because since as a cellular assay, you need to define how many cells I have to put on the plate. So for how long I have to incubate this assay to be able to put a new compound here and be able to kill the dengue, but not interfere with the cell host. But again, what our laboratory is doing is just a small contribution in the whole process. Drug discovery is a process that is long, is very expensive, and you have to work with many, many people together. And of course, this is very, very cost. So I just lost the battery here of the point, but so what you have here, so if you go on the right, on the green, you can see that the issue for develop new entities to approve new medications is something that take too long, take between 12 and 19 years, and costs more than $1 billion. So this is okay if you have time, if you are in normal conditions. But here in Brazil, in the last few years, you have suffered some problems with new virus, tropical virus that just show up, like you guys know about Zika, yellow fever. In this case, that you have something happen, you need to have, to be able to find a treatment in the short time. You cannot use the classical way, the 10 years, the long process. You need to try to find something in a very, very short time. So this is what I'm trying to do. You do in parallel. You're trying to do the repositioning of drugs. So what you do, you have 3 libraries of medications that are already approved to use in human, and you take these medications and you try on these assays to find something that is already too safe to use in human, but trying to see also if this can be used eventually in yellow fever. So it's clearly short, you can get this result in a very short time. You can get this result in the bench in few months. And working together with the doctors, I mean, you can give for them this option. So listen, at least in the cellular level, this can be an option for a patient that is dying. So when there is nothing better than that. Also, you see that for the cost. You reduce a lot the cost, about $500,000. So it's important to say to you guys, I mean, that at this point, I never have been worked with yellow fever before. I have experience to work with other viruses, dengue virus, chikungunya, but I never worked with yellow fever before. And this work was done by a PhD student in my lab, Denise, and Denise got this yellow fever, and using the technology and the know-how that you have in our lab, you got these results in 6 weeks. Everything that I go to show to you guys, she got this done in 6 weeks. So you need to be extremely pragmatic to be efficient. I mean, you cannot keep lost in time trying to define new things. So this is the picture that you take in the automated confocal. It's a machine that you use to screen. In blue, you guys can see the host cells. Use a human hepatocyte, HIL7. In yellow, you guys can see the yellow fever that was genetically modified with the yellow protein. Okay, when you see yellow here, is the yellow fever virus. As you can see, all the cells are infected with virus, but you see a couple of them on the right that are not infected. Unfortunately, I cannot show here. So what you do then, there is no physics here. There is no complexity here. So what you need to do, you need to teach the computer to find what is the cell. Because, I mean, remember this picture that I showed to you guys here? See here. Here you see infected cells with the virus. So you need to teach the computer what is the cell, what is the virus, because after all, we want to kill the virus, not the cell, to define the toxicity. So what you do then, you teach the computer to find the cells, the host cells. So how many cells I put in the first day, how many cells I expect to have after 3 days to calculate the toxicity, and use other parameters to define what is the virus. So the computer identify by the color itself, that is yellow. This assay is easy because I have 2 colors, was much easier. The computer identifies cell by cell the cell that are infected, and finally, the computer tells us that the green cells are infected. It is few red cells here that are not infected. Okay, so this is very important for us, I mean, because they bring 2 things for us. Once you establish this assay, you can do like thousands of compounds in a single day. It's already one advantage. You go very quick. And I think even more important than this, you remove the human being to be there choosing a good compound. Because when you have someone under pressure, they are tired. So what the computer do for us, just give some numbers, and you can define what is good, what is not good. You know, so remove the human from this. So what you have here, this is a scheme that show 1 plate of 384 wells, okay? And I have 1 picture for well, just to show for you guys have overview how it likes. So for this library specifically, I have 3 plates to cover the whole library, okay? This is one of the plates. On the right here, you have then hepatocyte cells, human hepatocytes that was infected with the yellow fever virus and was treated with interferon. So what interferon does, they clean the virus, okay? So you can see the blue that is the specific color that you stain the cells, okay? So if you have blue there, that means that you have the host cells there intact, but there is no more virus. So the interferon clean everything. So, and here are drugs that are using today to treat human, okay? In the pharmacy for different prescriptions. So what you can see here, most of the drugs, they are not able to clean up. They have no effect on the yellow fever itself. You see, most of the wells are very yellow. If it's yellow, the virus is there. Some of the drugs are toxic for the cell. You see here in black, the cell is not longer there. So what you're looking for drugs that be able to clean up, remove the yellow color, and left behind the blue. Okay? More blue I have here, less toxic. This medication was for the human cell. So this is the first screening, the primary screening. So in this point, what you do, you go back to our library and you test 10 different concentrations to make sure that result there is not artifact. And what she got, is here. So this is the representation of the screening, just for you guys to have idea. So this was a small library, but FDA approved library. And so what you have here in red, you have hepatocytes infected with yellow fever and not treated, okay? So closer to this corner here, more infected is the cell. And blue here, you have then cells that was infected with the yellow fever virus, but was treated with interferon. So in yellow, you have our compounds from this library here. Much closer to this control here, this is the candidates that you are looking for that can be eventually potential used in clinic, okay? So this is a different representation of the same screening. So what she found, and she has done all this work in 6 weeks. So this is very important, because you need to, when you have people dying, and I don't know if you guys follow what's happening here in Brazil, you have people dying here and people that was not covered because didn't take the vaccine. So you really see, there is nothing that you can do for these people. You be able to, at least to try and to offer some options. This is very, very important. This was very, very important for us. And what she found, she has done all this in 6 weeks. What she found that many of the drugs there on that library, that already, you see the prescription, the original prescription, from cancer to antibacterial. This is published. You guys can find this easy on the web. So drugs against different use, you found that they are also they have a good potential against yellow fever. These compounds that they have been described for some virus before, but not for yellow fever in that point. So have this essay working, a collaboration with Professor Dorigon and Professor João from Instituto de Medicina Tropical here in USP. We have access to sofosbuvir, as Professor Callas mentioned before, was a drug very efficient, against HCV. It's a treatment that is very, very expensive. So only this collaboration with doctors that work close to us that be able for us to have access for this. So, and you test this, so she quick tests the potential of sofosbuvir against yellow fever. So what you have here, you have then the red line. The red line is for the cell toxicity. You go to increasing the dose of sofosbuvir here, and you see eventually they become toxic for the cell, the human cell, but very light. And you can see, once you are increasing here the toxicity, you see the efficient against the yellow fever virus. So you have a very good window of selectivity. The selectivity in this case was around 300%. And even better than sofosbuvir is Harvoni. So Harvoni is from the same pharmaceutical company, the Gilead. Oh my God. Oh my God. Can you help me here? They're coming back, right? Ele vai vir sozinho ou não? Então aperta. Eu sou Macintosh. Ok. I'm a Macintosh person, so PC for me is very complicated. So what you have here, so this is for Sophos-Bovie. What you have here, Havoni, is a combination of Sophos-Bovie, and you see that Havoni, I don't believe have been used in the clinic yet, but Sophos-Bovie is even more promising because you have a very good, you see that it's very efficient against yellow fever, but more important, but something very important here to see the selective index of Havoni compared with Sophos-Bovie, nearly 700%, 700 times. Okay, so as a simplest contribution in a very complex process, the idea is, if you're not to be able to be competitive doing this, you have to work with people that is doing different things. So this is not the technology that I think this is more complicated part here. The challenge is to be working with people with different domains. And this is the biggest challenge. So I'd like to thank you then Europharma. I start with Europharma here. This is the biggest Brazilian pharma. Europharma is extremely ambitious. You see Brazil so far, the Brazilian pharmaceutical companies, they pretty much, they bet on do generics, on copy somebody else's ideas and make easy money, copying ideas from other companies, you know. So now this has changed because if you can copy a drug, the Chinese, the Indians can do better than us and cheaper than us. So the Brazilian pharma, they see they have to move for R&D. So Europharma, I believe, is that one that is doing more, is more ambitious on these directions, and Europharma is supporting our laboratory pretty much. So I'd like to thank then all my group. So it's a big group here. I have new members in my group that I didn't have time to put here. I'd like to thank Luis Carlos for support our lab, the head of department, Professor Márcia, and I especially thank you for Paulo Zanotto that hosts our lab. I mean, suddenly I didn't have a lab for some time, and Paulo hosts our lab. I mean, Paulo allowed us to work in his place. Professor Écio Dorigon and João Renato that have give us access to Sophos-Bovie. So thank you very much.
So open to questions. Yeah, speak louder, please.
Here, so in your system, have you used combinations, like you're using a polymerase inhibitor from hepatitis C? Have you used it with ribavirin or with other drugs and see if you can improve?
You did. You have this data. It's in this publication that I can give to you. You test men. So what I'm doing now, you have chikungunya, yellow fever, 4 serotypes of dengue. You begin to work now with Mayaro. So what you do then, you take all these controls that have been described for other viruses, and you test all this, and after you get what people already found out, I mean, this result was a group from Fiocruz got something similar to that, the same. So what you're trying to do, you're trying to see if the controls that you have is matching with the other groups, and since you are safe enough, you're sure enough, you go for these libraries, because it's so expensive to go to these libraries. But you have test this. I can make available. I can give to you the publication.
Did he answer your question? Hey, did he answer your question? Because his question is if you did combination of drugs to see if you could enhance the activity and have less toxicity. I think this was the question.
Yeah, so sorry, I didn't understand. So far, not yet. We're not doing this yet, but it's a way to go as well. It's a very good point. It's a very good point.
Yeah. Any other question? No. In this case, before the next presentation, our commander says that we have to take a picture. So everybody that's here, sitting here, has to come to the front. And they're going to take our picture. And it has to be now because there are many people that are leaving at noon. I'm sorry. Sorry, man.
Nick.
But, I mean, there is a commander here. Come.
Aqui a gente tem como esconder os ícones.
Muito bem, Luz.
Deixa mesmo com os ícones?
Se eu não fui claro, todo mundo é para vir para cá para tirar foto. Pour ceux qui parlent français, il faut venir pour prendre la photo maintenant. Désolé, mais on va arrêter la discussion scientifique pour une photo.
Finge que... Ah, na foto minha. Eu tiro tudo.
Aí.
Acho que dá para pegar aqui.
Acho que é melhor.
Hello.
Então me escolhe. Obrigado, viu? O microfone é aqui?
Está ligado aqui no verde.
So while you're sitting, I'm going already to announce our next speaker. Dr. Amaro Nunes Amato Neto is a physician pathologist at our hospital, Clínicas Hospital School of Medicine, University of São Paulo, and he has been very dedicated, he's very entrepreneur, has been very dedicated to study all the cases of yellow fever patients that died, and he's going to show us our autopsies, not the titer that is here. And many of those patients are the ones that Professor Esper presented in the previous talk. It's very interesting data he has, and I'm sure that with this kind of material, we can go very far in the pathology of the yellow fever. Amaro, thank you.
Good morning, everybody. Thank you very much for the invitation. I won't talk about sofosbuvir as the title says, but I will focus on the autopsy in yellow fever epidemic in São Paulo 2017 to 2019. I'm an infectious disease physician and anatomical pathology. I work at the pathology department of medical school. This is the building. In the basement of this building, there is the mortgage, the central mortgage of the capital metropolitan area of São Paulo. We perform the autopsies of all of the deceased people, patients from the hospital, but also from all of the patients that die in other hospitals that doesn't have any pathology department, they are not able to perform the autopsies, okay? Sorry, I've got a laryngitis in the last days. So why perform autopsies during an epidemic, okay? It's important for the preparedness. During the epidemic of yellow fever, at the beginning of 2018, it was installed a crisis committee in the hospital with a partnership with the Secretary of Health of the São Paulo state. All of the cases of yellow fever that were patients with, in a very severe conditions, were referred to our hospital. And so, during an epidemic, it's important for preparedness to understand the physiopathogens of a new disease, a new agent, an emergent or emergent pathogen, or a new strain of a very well-known pathogen with an unusual clinical presentation. So to understand what happens with this kind of cases in an epidemic, it's important to perform autopsies, to evaluate new therapies, to evaluate the quality of the medical services, and last one, but not less important, the differential diagnosis of disease that can mimic a certain epidemic disease. It's very important during an epidemic to understand what are the similar diseases that can mimic an epidemic. Our medical school has a transition to study the pathology of yellow fever. During the early 80s and early 90s, Professor Thales de Brito, a former full professor of our medical school, he described very well the electron microscopy of the liver in the yellow fever, and and he patronized an antibody that is used for our reference labs in all of the country, an antibody to search antigens of yellow fever in the liver and other tissues. He described in the early 90s that antigens of yellow fever are present not only in the livers, but also in the kidneys, in the heart, in other tissues. Professor Venâncio, he is one of our full professors. He is specialized, he is a liver pathology specialist. He published with Dr. Pedro Vasconcelos from Fiocruz, Belém do Pará, the criteria of visceral tropic disease, yellow fever vaccine, due to the vaccine. And in early 2000, Professor Juarez Quaresma from the Fiocruz, Belém do Pará, he studied the pathology of liver in the yellow fever with Professor Maria Irma Seixas Duarte, Professor Heitor, and Professor Maria Miriam Souto. They are full professors of the medical school department, pathology department. They described the in situ immune response in the liver in the cases of yellow fever. So the studies of pathology of yellow fever, the complete autopsies are very old. They study only the livers, and during this epidemic, we didn't know much about what happens with patients with yellow fever. For example, why they had convulsions, why they had so refractory shock, refractory acidosis, why these patients had pancreatitis. So that's why it was very important to perform these autopsies. And as soon as possible, we are submitting for an international journal. Until the end of July, this is 2019, we performed 85 autopsies with patients with suspicions of yellow fever. 7 of them were not yellow fever or other diagnoses. Among these patients, we had some extraordinary cases. For example, we have a patient that had previous kidney transplantation, so he was immunosuppressed. He had a contraindication to get the vaccine, and so he acquired yellow fever. He had fulminant disease. He lived in Mariporã, where was the epicenter of the epidemic. We had 3 cirrhotic patients due to alcoholism. So it is a chronic liver disease with acute damage, yellow fever, a tropical infectious disease. And in the international literature, it's much more common to have acute damage due to drugs. We have 4 liver transplantations, patients that were submitted to liver transplantation due to a fulminant hepatitis and yellow fever. I will show you the final results. It's published yet. 1 pregnant woman, 27 weeks of pregnancy, 2 adults with yellow fever, visceral trophic disease, and 2 children, 2 of them with suspicion of visceral trophic disease after vaccination. We are confirmed these 2 cases. Our mortgage, we perform about 15,000 autopsies per year. It's a lot of autopsies, about 500,000 from only the Hospital das Clínicas and the rest of hospitals in the metropolitan area. So we use during this epidemic old ancillary methodologies, optic microscopy, immunohistochemistry, special stains, but now we add new methodology with molecular biology to perform PCR in the organs that we collected, and minimally invasive autopsy. So we start to work with Professor Paulo Zanotto and his group, the LAMB, the Laboratory for Molecular Evolution and Bioinformatics, to study the molecular biology of the virus in the organs and also the host response. And the minimally invasive autopsy, a group that is headed by Professor Paulo Sadiva. We have this facility in the medical school, PISA, plataforma de imagem na sala de autópsia. Image platform in the autopsy room. We installed a tomography and a resonance and also ultrasound. And these are the other professors. And during this epidemic, we performed 17 minimally invasive autopsies guided with ultrasound. And after that, we performed the conventional autopsy. And we had the same results with the ultrasound-guided autopsy and the conventional autopsy in the diagnosis of the basic disease and also the immediate cause of death. And this article is under review in an international journal. So, revising briefly the yellow fever clinical feature, we have about 5 to 10% of the case in severe form with hemorrhages and fatal disease. So I'm going to talk about these cases, okay? Because yellow fever is a spectral infectious disease. Most of cases are asymptomatic or oligosymptomatic. And we are talking about this case, the top of the pyramid. So in the liver, we can find at the autopsy room, hepatomegaly. All of the livers has this color, orange and a yellowish color, because there is a icterus and also congestion. And after fixation, this is this color pale brown, and it's a very unctuous surface, cut surface, because of the steatosis. The main lesion is a mid-zonal hepatitis that affects the mid-zonal area of the liver, but also you can see a panlobular hepatitis because the patients had a severe form of the disease. Steatosis, it's these cells, for example, with a lot of fat droplets in the cytoplasm of the hepatocytes, and apoptotic bodies like this, the Kaussman-Rocha-Lima bodies. This is steatotic cells, looks like arachniform cells, and there is a lot of fat droplets in the cytoplasm. The pancreas, the results of the paper of Professor Callas showed that pancreatitis is one of the risk factors for death. So we found a ischemic reperfusion pancreatitis in all of the diseased cases of yellow fever. It's a severe ischemia in the retroperitoneal tissues, including the pancreas and the fat tissue in the retroperitoneal organs, with hemorrhage and steatonecrosis. This is a microscopy figure. You can see a vessel, arterial vessel with necrosis, ischemia, and an absence of inflammatory reaction different from, for example, the biliary pancreatitis, alcoholic pancreatitis, or viral pancreatitis for example, measles, or other virus. In the yellow fever, in these cases, we found only a ischemic hyperfunctional pancreatitis. This is a patient who had a fulminant course, had a very voluminous gastrointestinal bleeding. You can see here a ischemic and hemorrhagic necrosis of the stomach. This is a clamp trying to contain this bleeding, but it was not possible because the necrosis was diffuse with hemorrhages. The intestine has ischemic necrosis. This is very important because I think this ischemic necrosis is one of the factors that contribute for bacterial translocation across the intestine wall, and so the patient can have a bacterial sepsis. This is a vessel, a necrosis of the endothelial cells, and this is a vessel in the sorry, but the point is not. A necrosis, a partial necrosis in the endothelial cells in an artery in the pancreas. So this is a very important pathological finding in the yellow fever. You can see necrosis of endothelial cells. We are trying to understand better this process. In the brain, you can see congestion, cerebral herniation, and diffuse perivascular small hemorrhages. So this is one of the causes for convulsions and death. Some patients died due to cerebral herniation. And we are going to study what happens with these small vessels in the brain. Here it's a microscopy. You can see small perivascular hemorrhages, diffuse edema. And here, a hemorrhage around a small vessel in the brain. The kidneys. All of these patients, severe patients, had acute kidney injury. They needed dialysis during their hospitalization. And this is the basic lesion that you can find. In all of the cases, acute tubular necrosis and a mesangial cell hyperplasia in the glomerule. And something that was not previously described in yellow fever, we are describing now. You can see the bone marrow and also lymph nodes and also in the spleen. Hemophagocytosis, you can see a lot of macrophages engulfing some neutrophils, red cells. And so you can see an erythroid hypoplasia, megakaryocytes hypoplasia, amyloid retard, and hemophagocytosis in most of the cases. In the spleen, you can see an artery, and the lymphoid tissue around the central artery is completely depleted. So we can see in all of these cases a depletion of lymphoid cells. This is very important to understand what happens with severe cases because you can see in them an immunosuppressive state as they were a patient that suffered from a chemotherapy or offered even after a bone marrow transplantation. So we found severe infections in these patients due to bacterial nosocomial bacteria and also fungal infections mainly in the lungs. I will show you. This is the lymphoid tissue, very, very depleted. The lungs with congestion, hemorrhages, and this is a diffuse hemorrhage in the lung parenchyma. This is edema. It's very common in these patients. Severe hemorrhage, alveolar hemorrhage, and they died in the final stage with hypoxemia. And one of the most important findings, aspirative pneumonia with polymicrobial flora, with bacilli, coccus, and also fungus. And I think the lungs are a very important site for amplification of the inflammatory reaction that happens in yellow fever. It seems that the lungs is not an important organ in the physiopathogens of yellow fever, but I think with these autopsies that the patients After the fulminant hepatitis, they have a bronchoaspiration, develop a severe pneumonia, and so there is a bacterial sepsis that join to the viral systemic disease of yellow fever. And we found some cases with fungal infectious disease in the lungs, pneumonia. In this case, aspergillus, you can see the conidial head, and the IFA invading the bronchi and cause invasive pneumonia with angial invasion and dissemination in all of the bodies, committing other organs. So due to the lymphoid population, also antibiotics, and the systemic inflammatory disease, they acquire secondary infections. In the heart, you see some hemorrhages, interstitial edema, and the final stage of the disease are cardiac dysfunction. So what we already submitted and be accepted, the papers about yellow fever in autopsies. Submitted until now 5 papers, they are under review, and 2 of them are accepted. They are in the PubMed. The description of a case report, the patient with kidney transplantation. Dr. Ligia Sepiarotti, she works in the kidney transplantation department. And this paper in the histopathology described the 4 patients that underwent to liver transplantation. So we describe in this paper that after the graft reperfusion, the liver graft infect with yellow fever. So it's very important. I think it's one of the most important results in all of our autopsies, because we implemented in our hospital the liver transplantation for fulminant cases. And until now, I think 29 liver transplants were performed in the entire country, 7 of them in the Hospital das Clínicas, and others in Minas Gerais, Rio de Janeiro, Espírito Santo, and in the countryside of São Paulo. But I don't know if the other hospitals send the patients that died to autopsies to understand what happened. We send them, they are now published in the histopathology. You can see here, all of the livers in the 4 cases are very similar, quite similar with the other cases of yellow fever, with steatosis and icterus and congestion, and sign of ischemic necrosis. We see in all of the grafts the histological pattern of yellow fever with mid-zonal hepatitis, with apoptosis and steatosis of the liver cells. We found positivity for yellow fever antigens in the grafts. We performed the immunohistochemistry in 2 different labs. In our lab, in the pathology department, also in the Instituto Adolf Lutz. They were concordant. And in all of the 4 cases, the RT-PCR were positive, and also in other organs, such as kidneys, spleen, heart, brain, pancreas. So yellow fever is a systemic disease, and this is a kidney with a positivity in the tubular renal for yellow fever antigen by immunohistochemistry. This is a alkaline phosphatase technique. And this picture is in the paper, as you can see here. It's a proposal for the physiopathogenesis of the infection of the graft after liver transplantation. Probably the virions that is present in all the organs infects the graft, and the patient has acquired the disease in the liver, fulminant hepatitis, and all of the systemic process continues, and the patient dies from as if he was a patient with a yellow fever. So 3 of them survived in our service. Probably there is a resolution in some cases, but I think in most of them, they infect in the process, inflammatory process continues, and the coagulopathy, the vasculopathy, and the organ dysfunction, and so they die. So we have a lot of work ahead with yellow fever in our autopsies, with great perspective and expectations ahead. There is this quote from Marie-Françoise Bichat: "You may take notes for 20 years from morning to night at the bedside of the sick, upon the disease of the viscera, and now we will be to you only a confusion of symptoms, a train of incoherent phenomena. Open a few bodies, this obscurity will disappear. So this is one of the importance of the autopsy. For preparedness, diagnosis, epidemiology, to contribute to define all of the cases and differential diagnosis, learning, research, and of course prevent new deaths, because we understand the physiopathogens and offer better treatment after this knowledge. So this is a picture from Tarsila do Amaral, São Paulo. It was 1924. It's an exhibition in MASP until the end of July. I wish good luck for this partnership, and thank you. Thank you very much.
Questions? Yes.
So your data strongly suggests that you have extrahepatic replication of yellow fever. So how are patients treated prior to transplant, to liver transplantation? I mean, what's the parameter to say that the patient is okay for liver transplantation? Is it virus load? Is it virus in other cell types? What cell types are being looked at?
I think in this case that died, they had a positive, the RT-PCR in the blood were positive.
Fala no microfone, meu irmão. Fala no microfone.
The RT-PCR were positive in these cases. The other three that survived the RT-PCR were negative, but the determinants of infection or survival after the liver transplant must be better understood. And we have a small sample of cases to make inferences and affirm certain statements. But I think patients with severe disease in shock, in severe pancreatitis, and secondary infection, convulsions, we are not suggesting to transplant. I think the liver transplantation will be a measure that we abandoned in the future. We are in our hospital offering plasmapheresis that is published now in the PubMed. One of our doctors that leads in the intensive care team to treat these patients, they are working on plasmapheresis, trying to help these patients in the severe condition. Okay.
Any other questions? No? Okay, thank you.
So let's announce the next speaker. She's Dr. Tatiana Guimarães Noronha from the Fluminense Federal University in Rio de Janeiro, and she also doing some collaborative work with Biomanguinho as well.
Yes. I need to put here, or have assistance? Oh, sorry. Good morning, everyone. It's a pleasure to be here. I'm representing the Biomanguinhos Fiocruz, where I have been working in the last 15 years in the clinical unity. I also work at Universidade Federal Fluminense, but I also work for a long time at Biomanguinhos Fiocruz. I'd like to thank for the invitation, and I'd like to present to you some results of the clinical research that have been conducted by the collaborative group on yellow fever vaccine. The time will be a big challenge, but I will try. Just to remember, the yellow fever vaccine, Dr. Akira have already exposed a lot of this vaccine. It's attenuated virus vaccine used in Brazil since 1937. This vaccine has a well-established effectiveness. With these vaccines, it was possible to control yellow fever outbreaks over wild in the last time, and usually in the currently it's possible. It has the no reactogenicity profile. I'd like only to highlight this information in red about the WHO requirement for vaccine potency. The minimum requirement is for each dose is a minimal 1,000 LD50 or equivalent in PFU. In Biomanguinhos, our control quality department have done this work for all lots that for the commercial lot. In our evaluation, we have the commercial vaccine produced by Biomanguinhos have in average 6.6 times above the minimum requirement for yellow fever. This information is important for the next slides about the data related to fractional dose. About the yellow fever vaccine safety, only a brief overview. In the last years, there were related about the severe event adverse, mainly the visceral-tropic event and neurological effect event. This information gave to us some concern about the safety of the vaccine and indicated some studies about these issues besides the national surveillance about adverse event. Only a brief related about the neurological disease. We have two kinds of neurological adverse event, the neurotropic, more severe, and the autoimmune disease. Data from Brazil are related one case for 500,000 doses applied. In Brazil, we have more recent data, not published yet, and with the last 2 years, since 2017, when we have increased in vaccination in Brazil, there is not difference from this rate that we are observing in the last years. About the visceral tropic disease, the more severe case, the more concerned case, it are related to the first dose in our reality. Some factors have been related for the causability with this event, but it's individual host factors. There is some contraindications at immunosuppressed patients. There is some controversy about elderly, and in the last study observed that it's not the relation with the elderly with high risk of the adverse event. In our national data, we also can see in the line of 60 or more, there's no the risk relative is not defined for the elderly. This is a recent systematic review published recently. It's conducted for independent group from Biomanguinhos and Fiocruz. There's the conclusion that I put here in the intergreen of the manuscript, and they confirm the safety about the yellow fever vaccine, including for the group risk that we have considered for no indication to vaccinate. They didn't saw any relation with this kind of conditions. With the lack of alternative explanation about the serious adverse event, we are conducting a study in partnership with Rockefeller Foundation about genetic factors that could explain the occurrence of this kind of event. In a manuscript published yesterday, we have a preliminary analysis of a deficient one factor related to interferon production that is related in two cases of severe yellow fever, severe adverse event to attenuated vaccine. In one case for measles vaccine and another case for the yellow fever vaccine. So we are already conducting this study in Brazil for any kind of severe or of visceral tropic disease. We have a partnership with the Ministry of Health, the surveillance of Ministry of Health, to get access to this case and get biological material to continue for this kind of evaluation. We agree that this information would be useful in the future for the indicate or not indication vaccinates in some group of population, particularly in the parents or another kind of relation, genetic relation with the case of a severe adverse event. About the immunogenicity of yellow fever vaccine, we don't have a correlate of protection in humans, but we have values of some markers, neutralization antibody by PRNT, that is useful in the immunogenicity study with this vaccine. We know that for adults, the immunogenicity is higher than children, and especially in children vaccinated lower than 2 years of age. For the usual dose of yellow fever vaccine, we get less than 90% of seroprotection 30 days after the first dose in these children. This information is related with the two kinds of discussion that we have with yellow fever vaccine. That is the duration of immunogenicity that has impact on the vaccine schedule, and the use of fractional dose, the recommendation for fractional dose in an emergency situation. Here, just to remember you, the recommendation from WHO in 2013 that the yellow fever vaccine could confer sustain lifelong protection after only 1 dose. This recommendation was for all the world. In that time, in our national immunization program, kept the recommendation for the boost dose, at least in the children with 4 years of old, until we have epidemiological situation in the last weeks and the high demand of the vaccine. During this time of this publication from WHO, we are conducted some studies, Fiocruz, with partnership with universities, with the army in Brazil, with secretaries of health in municipalities in some states. We were conducting some studies about duration of immunity in the regular dose, the usual dose of yellow fever. We had one study in adults. After 1 dose of this vaccine, stratified 4 times since the vaccination, in adults 2, after 2 doses, and in children vaccinated in the first 2 years of life. The study in children is not already published. It's a doctoral degree thesis, and we have already submitted to the publication this month. Here, a brief summary of this data for adults vaccinated with 1 dose, regular dose, usual dose, recruited in the states Rio de Janeiro and Minas Gerais. They received the first dose, and it's a retrospective study when we evaluated the immunogenicity for proportion of seropositivity and geometric mean titer since the time that last the vaccination. We can see that the adults vaccinated from 5 to 9 years ago, there was a drop in a decline in the proportion of seropositivity until 83%. In the adults vaccinated after 2 or more doses, we have only 8 that have read more than 2 doses. The big part of the sample size have received 1 dose, and they were analyzed considering the time since vaccination. And we can see even for that received 2 doses in the past, there was a decline in the seropositivity rate from 6 to 9 years after the vaccination. So we understand the public health conditional about the indication that 1 dose or more. We understand the concern about the shortage of the vaccine, but this study can induce the evaluation of the necessity of the booster dose, at least 1 booster dose. And in children, that group that usually has seropositivity rate lower than adults, Just to remember, for usual dose, 30 days after the vaccine, almost 87% is the group that is considered protective from the vaccine. When this group is analyzed years after the vaccination, we can see that the seropositivity rate declined approximately from 2 years, since 2 years after the vaccination, for 31%, and this decline is more accentuated after 4 years after the vaccination. About those response study, just to remember, the first study that is related is 1940. This study gave us the information about the minimal immunity dose for this kind of the vaccine. After, in 1988, Dr. Souza Lopes had a study for those response study. It's the first study that indicated that the lower dose that usually used would be enough to get protection to yellow fever. They saw that doses from 2,000 to 200 PFU, the units of that time that usually use that time, would confirm immunity to yellow fever. But this study, there was question related to sample size. It was not possible to change the indication with this. So in 2009, Biomanguinhos, after requesting from Ministry of Health, because we have in Brazil in 2007, 2008, episodic increase of case of yellow fever in Brazil, and the Ministry of Health requested us to improve our capacity to produce the yellow fever, and request us the continuous study about those response. The question if it's possible to reduce the dose of yellow fever vaccine to guarantee the same protection and to improve the capacity of production by Biomanguinhos. So Dr. Reinaldo led this study. We studied the usual dose with the unity of almost 30,000 international units of the vaccine and 5 decreased dose, approximately 3 times lower, 10 times lower, 50 times lower, 200 times lower than the usual vaccine, and also 1,000 lower than the usual vaccine, the commercial lot. Here we can see the results. Doses down to 500 international unity, approximately 200 times lower than the regular dose, the commercial dose, they had the similar circulation rate, the similar immunogenicity, considering the reference dose, the first one. After 3 days after the vaccination, there was not difference between this concentration until 600 international unity. This subject had already been evaluated 10 months after the vaccination, and they kept this protection except for that group vaccinated with the 31 unity international 1,000 dose, 1,000 lower dose than the reference dose. With this study, the WHO, using especially this study, the WHO recommendation, the fractional dose, there was a difference between our study in the past and the WHO recommendation. That study was done with the lot produced by quality control. It's not reduced during the application of the vaccine. It's a dose contained in 0.5 milliliters of the vaccine. But they use this study as information that could use the fractional dose as an emergency in public health. So they recommended this in 2016. These strategies were used in Congo, in Africa, and we have already the manuscript, the paper published from that group with WHO, demonstrating that the possibility of control, the fractional dose in control outbreak in that situation. In that time, Dr. Reinaldo was the member of the WHO group, and considering this possibility, they had the idea to recruit again the same subject that participated in this study in 2019 to understand, to know how was the immunogenicity of yellow fever 8 years after the dose response study. 8 years because its study have been done in 2017 and the vaccine have been applied in 2019. As we did this kind of evaluation was not planned in the original protocol, we need to recruit again for these volunteers. We cannot have the 1,700 subjects that already participated in the first time. We have approximately 317 subjects. We excluded those who have been vaccinated after the dose response study, and those have related travels to endemic areas or another kind of vaccination or living in the endemic areas for yellow fever vaccination. And we can see here for the proportion of seropositivity, we have the same there are not difference between the group for the proportion of seropositivity 80 years after the vaccination. Regarding the geometric mean titers, we have a significant for the one group, the 31 group. The group, curiously, the group that received 1,000 times lower the dose, but we have here a small group because the group have already been revaccinated for us after the first time of the study. Here we have a statistical impact in this result, and maybe the capacity of this individual to well respond to yellow fever. Just remember that they were able to keep this immunogenicity 30 days, 10 months, and 80 years after immunization. So there is some discussion about the genetic factors in this kind of answer to the vaccine, and we are studying this yet. Just to compare the data that we have from the duration of immunity for the group that receive usual dose, here we had the analysis considering the same cutoff that another study that used, and we have the similar compartment here in the letter B is the current study, 8 years after the vaccination, and in the lines above is the first study. There is not difference in the proportional seropositivity and geometric mean titers 8 years after the dose response study and the usual regular dose. We have also the cell immune studies conducted by Instituto René Rachou, also from Fiocruz. In the same samples, in the same study, Biomanguinhos conduct the humoral evaluation, and René Rachou the cell immunity, and the results are similar to our study. Our current and plan study, now we are recruiting the same volunteers from the study of 2019 to conduct an evaluation 10 years after the dose response study. Our plan is to conclude this kind of analysis for dose response until 10 years after the vaccination. The study is now, currently we are in the phase of recruitment and blood collection. We also have ongoing the humoral and cellular immunity and subjects that receive the fractional dose in the campaign in Brazil. And this study is already ongoing. we have, and the first one, I confused, is a 10-year cohort study that we are conducting in Paraíba with more than 4,000 subjects that will be followed for 10 years. It will be a prospective cohort study to confirm that our finds about the duration of immunity. Here we have children and adults vaccinated with the first dose and followed by 10 years. We are also planning the dose response study in children, the same design to the adult dose response study. We have already funding for these studies from WHO, and we are planning also, it's under discussion yet, the duration of immunity after 2 doses in children vaccinated in the first two years of life. Here, in special announcement to Dr. Reinaldo, I mentioned here sometimes in these presentations, Dr. Reinaldo led in the last years, more than 15 years, the study with vaccines study in Brazil, especially for yellow fever vaccine. He is a reference, a national and international reference in the issue. And it's here an interview that he had. He passed away in the beginning of this year, and this interview that he done to our about comparing with the Institut Pasteur and Oswaldo Cruz. He has working in the conclusion of analysis of 8 years and protocol for 10 years in those response to children, even during its hospitalized, and Oswaldo Cruz and Pasteur, they were the inspiration from him, to him. And also, my last slide, the collaborative group for study with yellow fever, Dr. Akira, Dr. Ricardo Galler, Dr. Reinaldo, and some key persons. The group is a big group that have been working the last years about this vaccine, but here Dr. Camacho, Lourdinha, Dr. Olindo, Dr. Marcos Freire, key persons from clinical research, from lab, from design, planning, and analysis for this study. And many thanks.
Thanks, Dr. Tatiana. We have time for questions. We have about 15 minutes or so. So if you have questions.
I have a question. Thank you, very interesting. Lots of interesting data. The question would be, do you think that antibody is a good biomarker of protection? Because, in vaccination, we always look at antibodies. And I wonder if this is a good correlate, actually. Could you approach this question for me? And what would be the titer that would be protected?
So the antibody titers, the neutralizing antibodies, the more specific antibodies, as I have told you, we don't have the correlation of seroprotection in humans for yellow fever vaccine. But it's a vaccine that has been done for some decades. And there are some data with the correlation between the effectiveness of the vaccine, the possibility to control outbreaks, and antibody titers level. So it's a reference, international reference, including for WHO, about the immunogenicity of yellow fever. I agree that it's not enough. For this, we are continuously studying the cell immunity associated, but it's the biomarker that we have until this time, and this but I agree that the cell immunity has a big importance of these kinds of evaluation. And we use the PRNT with the methodology. For particulate of the metal, there is a big variability between the labs. You use, for duration of immunity in the adults, 2.7 logarithm, for the study in dose response 2.9, but there is, for particularity of the method, there is a variability between the time of evaluation, but the test is validated according to international requirement.
Yeah, I ask you because all the decisions done by WHO and other people are based only antibodies titers.
Yeah.
And sometimes, perhaps now have much more, tools to study other types of immunity that could correlate. As I've mentioned in the beginning, I think, of the session, patients in our cohort of immunodeficient patients that don't produce antibodies, they could receive the vaccine and they didn't die. So they have to eliminate the virus some way. They didn't have the visceral disease, as Amaru showed us, for other individuals that normally produce antibodies.
Yeah, the neutralization antibody is a particularly known that a good, it's not a correlated, but a surrogate of protection. And that's the information that we have at this moment.
I agree. I just wanted to bring the question.
Do you think there is some change in volunteers with dengue fever historic infection in areas where dengue fever is endemic?
Yeah.
The response in that individuals?
Yeah, because unfortunately we don't have enough time here to present the overall data, but for each study we have done evaluation for dengue before vaccination, after vaccination for those response studied, and the observational cohort for duration of immunity. We will also analyze the dengue. At this time we didn't have Zika yet in Rio de Janeiro and in Minas, but we did the analysis, considered the dengue antibodies, and there was not the relation between the result with the immunological status for dengue.
So in the same line of marker of protection, the serology that's been performed uses the vaccine strain, am I correct? So do you think we may find different results if we switch from the vaccine strain to the wild type virus when you do the PRNTs? In other words, this could result in different findings in terms of neutralization capacity after vaccination.
Yeah, we have some study. The Fiocruz, Instituto Oswaldo Cruz, made a study about the strain of the virus, and the kinds of mutation that have been found is not related to the vaccine action, and neither for the neutralization antibody. So we don't expect that the difference to the response to vaccine with this mutation that already been found in the last time. I don't know.
But are these side-by-side being conducted? Have you performed validation comparing wild type and vaccine strains?
Yeah.
Okay, thank you.
If there is any question, then you can address any of the three speakers or comment.
In yellow fever.
On yellow fever, of course.
So, no more questions? Okay, so thank you very much. Good lunch. Thank you.
Thank you.
So, thank you, everybody. And now we are going to start with the roundtable 4. And the idea is to discuss and present the idea towards a bioscience network, AMSUD-RIIP. And so the idea is that Otto Pritsch from Institut Pasteur Montevideo is going to give us an introduction about the things that were already done in this project. Then we are going to present the Association of National Universities from the Montevideo group. And finally, we are going to mention two actions that are going to be taken, are going to be happen at the end of the year in Bolivia with the metagenomic and bioinformatics approach. And the French cooperation is going to tell us some news.
So, okay. Thank you, Carlos. I think that, okay, this is the plan of the roundtable. As Carlos said, I will do a brief introduction. We think to have here Álvaro Maglia, which is the executive secretary of the AUGM, but he's blocked in Portugal. Álvaro Maglia, who is the executive secretary of the Association of Universities Grupo Montevideo, can't come here. He is blocked at Portugal airport. He will be here this evening, and Carlos will present the association. After Alexia Gobrecht from the French Consulate of São Paulo would present the organization and action of the French cooperation in South America. And Gregorio Iraola and María Teresa Álvarez, Gregorio from Institut Pasteur de Montevideo, María Teresa Álvarez from the University Mayor de San Andrés de La Paz, Bolivia, will present the organization of two courses that will be organized this year. We have initiated this cooperation at Pasteur Institute Montevideo with the organization in November 2016 of a course focused in the bioethical aspect of research in human genomics with the cooperation of the French cooperation, UNESCO, Institut Pasteur de Montevideo, de Organización Panamericana de la Salud, and FOSEM, which is the Fund of Convergence Economique du Mercosur. This course have as a professor two French professors from CERN, Hervé Neuweis and François Hirsch, and also professor from the Sociedad Chilena de Bioética, Manuel Santos Alcántara, and Víctor Penchasade from UNESCO. The second course that was organized by Pasteur Institute of Montevideo with the cooperation also of French cooperation and FOSEM was a course focused on hands-on metagenomic data analysis, specifically to work with tools for bioprospection in clinical and environmental microbiology. This course that take 1 week in Montevideo has a satellite seminar in Buenos Aires organized by CONICET, the program franco-argentino de seminarios. Sorry. Yes. In June 2018, at the University de la Republic of Uruguay, it was signed an agreement between Institut Pasteur de Montevideo and la Asociación de Universidades Grupo Montevideo. This agreement was signed in the rector office as a corollary of a series of approaches that began in 2016 with the participation of the French cooperation for Latin America. We have here in the photo Luis Barbeito, the former executive director of IP Montevideo, Ricardo Ehrlich, the president of the Conseil d'Administration de IP Montevideo, and the rector of the University, Marcarian, and Álvaro Maglia, the executive secretary of the association, and the presence of Leticia and Marie-Christine, representatives of the French cooperation in América du Sud. What is AUGM? It's a network of public universities of Argentina, Bolivia, Brazil, Chile, and Paraguay, and Uruguay. It was created in 1991. It constitutes an expanded common academic space of scientific, technological, educational, and cultural cooperation. The 39 universities that integrate it have the condition of being public, autonomous, and separated, self-government. Last December 2018, a third course was organized at Institut Pasteur de Montevideo, focused on hands-on microbiome data analysis, with focus on tools for understanding microbial communities in health and disease. And this was the first course that was co-organized by AUGM, and we have a picture of for example, the web page from the Facultad de Farmacia y Bioquímica de la Universidad de Buenos Aires, who present the course for all the students of the fac. At this moment, at Montevideo, it's held a technical meeting with the support of the French Embassy in Uruguay and the French Regional Cooperation, in the presence of the representative of the Institut Pasteur de Paris, principally from the bioinformatics department, the Z3BI, the AUGM, and several South American partners, Argentina, Bolivia, Brazil, Chile, Colombia, Peru, and Uruguay. And we have signed a proposal for the constitution of a biosciences network by the participant institution. From Argentina, it participated CONICET, Ambassade de France en Argentine. From Bolivia, la Universidad Mayor de San Andrés de La Paz. From Brazil, la plataforma científique Pasteur USP, l'Ambassade de France au Brésil. From Chile, el CONICET. From Colombie, la Universidad de La Sabana, l'Ambassade de France en Colombie. Pour la France, la Délégation régionale de coopération pour l'Amérique du Sud et le département de bioinformatique de l'Institut Pasteur de Paris. Pour le Pérou, l'ambassade de France en Pérou, et pour l'Uruguay, l'Institut Pasteur de Montevideo et l'ambassade de France en Uruguay. Et aussi des représentants de l'association de l'Université Groupe Montevideo. Je continue en français. Je suis comme les consuls. Sorry. The general objective of this network is to promote scientific collaboration between South American and French academic institutions in the biosciences area through support for development of scientific, technological, and innovation capacities, the formation of local resources, the cooperation and mobility between researchers from the region, the collaboration in publication and other form of intellectual production that allow advance in the disciplines of common interest. A second general objective is in a first stage and in a consensus between Pasteur Montevideo and the Département de Bioinformatique de l'Institut Pasteur de Paris. It is proposed to prioritize the area of bioinformatics without prejudice to develop other areas of interest in the future agreed by all the participant institutions. This initiative has the vocation to join other regional efforts that are developing in the same direction. And now, Carlos will continue with the presentation.
Thank you a lot. So now the idea is to tell everybody about the Association of Universities from the Grupo Montevideo. As we mentioned before, there are 39 national universities, public universities from South America that are together in a group that is called the Grupo Montevideo. So, excuse me because I'm just trying to help Álvaro, who is delayed in Portugal. So, what about the numbers that are in the association? We have around 3 million undergraduate students, more than 400,000 postgraduate students, and 200,000 professors and researchers between all the 39 universities. Over than 2,500 undergraduate, postgraduate, and professors are going from one university to another each year. So when it was like 30 years ago when the association was established, the main purposes were to have like a critical mass of qualified human resources, taking the advantage of all the capacities already installed in the region, trying to have strategic areas in common from a scientific, technological, and innovation point of view. Also, one of the main purposes of the association is to continue training and improve the capacities of our human resources and try to improve the interactions between our members and our societies as a whole, okay? So, and one thing that is in common with Luis Pasteur is one of the ideas was to spread the knowledge, helping with the teaching and spreading the knowledge with the society. So, the programs that are being supported by the network are from a scientific cooperation. We have different groups going from social areas to life science areas and academic committees. Marta is one of the coordinators from the Human Health Committee from the University of Pharmacy and Biochemistry in Buenos Aires. Also, we have a mobility of a lot of students, undergraduate and postgraduates, and also different researchers and professors. Summer schools are always being held, meeting of young researchers, and different networks between cities and the universities. Also, there is we are trying to join the university with the states and with the society to address different important public problems or questions that arise. So how is the governance and the financing of this important association? So all the universities are public, non-profit civil associations. We have a council of rectors. Each university has its own rector. So we have the council of all the rectors together. Then we have a president and a vice president that are being elected between the council members, a secretary executive, Álvaro Maglia, and a group of advisory delegates. All the universities are self-financed, and the cost of mobilities and programs are covered by each university. So the budget is very difficult to estimate because it's not cash budget, but it's kind of in-kind budget with around $5 million per year. And then we have permanent commissions, and I'm highlighting the postgraduate and science, technology, and innovation commissions, where the problems are being addressed and analyzed and studied between the experts in those fields. I think that's it. Yeah. So Alexia is now going to tell us about the French cooperation.
No. Oh, okay. You want to talk about the partners? Okay. Okay, thank you, Carlos. So yes, the idea is to present a little bit how the French regional cooperation in South America works as a network. So as I'm working at the French consulate in São Paulo, I'm just part of the piece of the network, but the coordination of this network is held by Leticia Chilicini from the Delegation of Regional Cooperation, which is based in Chile. And unfortunately, she couldn't come here today for this meeting. So first of all, you have this delegation of regional cooperation, which works with all the countries of South America. As I said, it's based in Chile, but has link with all the different countries through goes too fast, to the cooperation services that are embedded in all the French embassies of the different countries. So these services are headed by a counselor of cooperation and cultural actions, which we call the COCAC. And the point of contact of every action that are implemented in this cooperation service are the attachés. So you have scientific attaché, you have cultural attaché, etc. So this is the network. It's not working. It's okay. So, which is important to know is that there is a strong relationship between the priorities that are proposed by the delegation of the regional delegation and the local point in the embassies. So these priorities can be, in the field of scientific cooperation, governance, human rights, cultural cooperation, influence, and intellectual debates. So all these priorities are proposed to the local point, the local embassy, and of course also with the objective of the Ministry of Foreign Affairs in France. And regarding these priorities, the embassies and the cultural service will suggest a programmation of action regarding these priorities locally. And finally, the delegation, the regional delegation, will coordinate all that action. Which is important to stress out is that the implementation of these actions supported by the regional delegation will be held by the local service in the embassies. And also, also the communication of those actions. Okay. So to give some examples, I will give a little bit the priorities regarding the scientific cooperation. First, the regional delegation will organize and spread information and coordinate what we call the France-South America Joint Program. So maybe you know STIC and Math AMSUD. There are projects called PREFALC. There is also a partnership with AURM. These programs are mainly dedicated to academic exchange, research projects between at least 3 countries of South America. And regarding this program, there is also the regional network in bioscience, which we are talking about here. The second point, the delegation, the regional delegation can develop new programs. For example, this year there is a new program about climate change issues called Climate AMSUD. So the regional delegation is at the starting point of this new program. So what was the role of the French cooperation in this regional network? So as Otto said, the starting point was the initiative of Institut Pasteur Montevideo and the AUGM, which is so in Montevideo in Uruguay. So there was a strong support of the local embassy, of the embassy of Uruguay, the French embassy. But then when the idea came up to spread through first the network of the AUGM and to spread the action and the courses, for example, to other countries, the regional delegation could support this initiative because it works really in all the countries of South America. Therefore, after the meeting of last December, with these good intentions of spreading this action all over South America, the French delegation supported 5 actions for 2019. So the first action is, we are in the middle of it. So what did the regional delegation do for this scientific symposium is to invite 5 researchers of South America. So we have Marcelo, we have Marta, we have Jaime, we have Cesar and Gregorio, who is here, and who did a class on Tuesday about metagenomics. And so the idea is to give a first link with these universities in which these researchers are working, and so we were thinking it was a good idea. And we also thank the RIIP, because I think it's, I don't know where the RIIP organizer, but it's also a little bit new for them, which I was suggesting. It's new for them to include other partners than only the RIIP partners in the scientific symposium. So in Colombia will be held immunology and immunogenetics course this year. In Argentina, like last year, there will be the course about metagenomic. And in Uruguay and Bolivia as well, there will be this year also bioinformatics course. And so the idea here is to give the hand to Gregorio and Maria Teresa, who implemented this action in their university. Just to finish, the support of the French regional delegation for all this action is about 27,000 €, which is quite a lot. C'est tout. So it's an important action for the French cooperation to support this network. Thank you. This is Gregory. I think you have to. I will. You want to? Oh, Maria Teresa. Maybe you can. Ah, Maria Teresa. Okay. Carlos. Oh.
No. I think it's yours. Yeah. Okay. Very briefly, since last year, based on the interesting meeting that we had in Uruguay, where the Bioscience Network was established, researchers from the Institut Pasteur were invited to cooperate with Bolivia to organize a course, a postgraduate course, or courses from now on, and later on perhaps to establish some projects based on common research lines. Where is there? Begin. Bing. Yeah? Oops. Yeah. Okay. And then, in general, in Bolivia, we have several groups, research groups, but in special working in microbial genomics, we have the demand to enhance skills and expertise in the management and analysis on metagenomic data to identify genes and functional genes for medical and biotechnological applications. That's the main and the basis for to propose this course, and the call was on the internet during June, and it was the basic tools on bioinformatic applications in metagenomic. And the deadline was the June 30, and we have 56 postulations now on, and then we have in the process of selection. Then the objectives of the course mainly were the knowledge transfer about the new trends about genomic analysis, to assure quality based on the expertise of the researchers of Institut Pasteur, to strengthen the relationships between Bolivia and Uruguay, perhaps later on, to establish some relationships based on our proposals. And then, since we are now in the selection process, we have a lot of work to do during these days, because we have to answer in a few days until July the 10th. And then in September, we have to offer some support material to our students. Perhaps we can facilitate some special computers, or they have to carry the computers to facilitate the Linux and other programs like R and Python. Then after the course, we have for sure the evaluation, and then if everything is okay, we can continue with this kind of courses for the future. And perhaps for future perspectives, we can continue with we can join forces to establish some research projects as well. That's all I have to say. This is your.
It's the last one. Last one. Oh.
Okay, I will briefly tell you about our experience on developing these kind of courses in Pasteur Montevideo. We already had 2 courses, one in 2017 and the other one in 2018, and we are already organizing another one for this year in December. So basically, our approach for these trainings is to perform what we call hands-on courses, in which students spend most of the time in data analysis sessions, and the other part of the time are lectures or something like that. But the first one we had, that was this one in 2017, was a 2-week course. And in particular, this was really interesting because the students brought their own data for being analyzed during the course. So this was a really nice experience, but at the same time it was really challenging and time-consuming for us, because we had like 25 students, each student with their own data set, trying to solve problems regarding their PhDs, because the data was data that they were using for their thesis or something like that, or for their papers. And in one hand, it was really interesting and really motivating for the students, because they felt that after the course they advanced in their own projects, but at the same time for us it was very very challenging trying to answer all these kind of different questions. And another really nice thing that we achieved with this course was that in collaboration with some ambassadors from the American Society for Microbiology in Uruguay, we were able to perform live streaming of the lectures of this course to the rest of the Latin American countries, and even for countries in other places of the world. For example, one day we had one speaker, and we had people from Hawaii connected, looking at the speech of that lecturer. And in total, we had over 100 people connected during the course from different parts of the world, watching the lectures from our invited speakers. So this was a really nice experience that we hope to reply during this year. So we could expand the audience of the course in a very, very interesting way. In general, we perform this for more or less 20 to 25 students, because it's the size of the room that we have for computers, but also it's a number that we can manage. And in general, we have around 2/3 of the students are from the region or international, and another part are from Uruguay. The second, this is not working. This is just as I don't know if you can see, but this is like a summary of the schedule of this first course during the 2 weeks. And as I mentioned before, it was around 20% of the time were like lectures from speakers, but 80% of these 2 weeks were data analysis sessions in which students were fighting with their own data. So it was really, really nice to see this. I can go ahead with this. This was the course in 2018. After that course in 2017, we decided to shorten the course to 1 week and to organize this one in particular with toy data sets, not with data from the students. So this was a shortest course, but more focused on a particular topic, that was the analysis of microbiome data from different situations in which we can compare disease and health conditions. Also was a course for about 25 students. So in these 2 years, over 50 students from different countries have participated directly on the course, and we had more than 10 speakers and instructors from different countries, in particular from Paris, and also from Latin American collaborators, and from other countries like the UK and the US. Okay, and this was just to say that in this course in 2018 was the first time we performed some diffusion in collaboration or together with the Asociación de Universidades Grupo Montevideo, and also that I think that was mentioned is the course in Bolivia that is in this context that is going to be developed next September. And the last one is the upcoming course in Montevideo in this next December during the first week. We didn't launch the applications yet for this, but information is going to be available very soon. And in particular, this course is going to be focused on applying genomic and metagenomic tools or approaches to analyze sequencing data to answer questions regarding antimicrobial resistance. We have some already confirmed speakers from the Sanger Institute, also some instructors from Pasteur Paris are coming, 2 of them, we don't know the names yet, and also from Argentina, Josefina is coming to help us. And of course, this is going to be in collaboration with all these institutions that you can see there. And more or less, this is what I wanted to say, and the general overview of the teaching activities and training activities that we are developing, and hopefully we will continue developing this kind of courses in the future. Was very important the financial support that we have received in this period from the French Cooperation Agency and also from the United Nations University in its Latin American and Caribbean program, the UNUBIOLAC course. And of course that and the Pasteur Institute for all the support. Yeah.
So now we will have time for discussion, and so one thing that you may have noticed is that we have learned in the last few years in Montevideo is how to make Gregorio to work. So if anybody needs a course, we can send Gregorio. So these are the big points that we want to address and to discuss. We have time, so if you guys want to make any comments, questions, whatever.
Sorry, no, one thing that I forgot to mention is that after every course, we, in collaboration with the people in Buenos Aires, we develop a satellite event that is like a seminar that is focused on a certain topic. For example, past year was about genomics and health. So we bring the speakers that we have in the course to Buenos Aires for a whole day of scientific talks for different people in Argentina. We had done this 2 times, and probably this year is going to happen again.
Okay, thank you very much for this very interesting presentation. Can you please tell us the percentage, for example, for this last course, the percentage of students from
Uruguay, and the percentage of students for different countries of South America. That will be very, very interesting for us. And what will be also interesting to know, as you mentioned this, for these courses, for example, there is many organizations that help for founding the course. What also the percentage of how it's distributed? That will be also interesting for all of us.
Sure. The last course, for example, were like more or less 10 Uruguayan students and 15 foreign students. This is more or less the rate. Is always is over 50% students from other countries, and most of these students from other countries are from the region, from Latin America, basically, because during these courses we pay the accommodation and meals for students during the whole course, and also we provide some travel grants. So we can't provide travel grants for people that is living too far away from Uruguay. So most of travel grants that we pay are from people that is close to Uruguay. So this also bias a bit which countries we can fund for students to come. And the other question that was regarding the funding, these courses to cover the whole operative costs are around $25,000 to $30,000 per course, including all these things. And for example, the UNU Biolac applications for courses in the Latin American region provides $50,000. And the rest is between basically the Pasteur Institute and the French Cooperation Agency.
Thank you. Then the courses are in English,
All in English. Yeah.
Then the majority of these courses are focused on students comes from South America. But what about the students come from other an institute of the network, of Pasteur network?
Yeah, that's very good question. Indeed, for the course, we already identified this problem because we were receiving a lot of students from South America. That was really nice because we wanted to strengthen the training in South America for Latin American students, but also we don't want to prevent students from the RIIP, for example, to come. So we identified that problem, and for this course in this next December, finally we could obtain some extra money from Pasteur Paris that will pay for some travel grants for students within the network. So we will pay the grants, the travel grants from the local and regionals, and Pasteur Paris is going to provide the money to bring the speakers, but also to bring 4 or 5 students from the international network.
That's great. How you diffuse the information? How you say how you diffuse how you promote the course? I mean, are you doing that across your website, or do you have another network to do that?
So, an important thing is that because now the Pasteur Montevideo Institute is part of the Joint Association of Universities in South America, we send information, and each university is also spreading the information. So that's an important way, an easy way to reach almost 3 million students from the region. Okay?
No.
You diffuse also in the...
We also send information to the RIIP. Yeah.
Yeah, it's the last courses have appeared in the RIIP newsletter and also in the web page in the Pasteur Paris web page, and also Antonio Borderia is the guy that is in charge of that and working with us for diffusion along the RIIP.
Okay. I have last question for Alexia. Alexia? Yeah. Yeah, then you mentioned that you have 27,000 euros for the cooperation. Is that consecrated only for scientific cooperation or for all the cooperation that managed by the office in Chile?
No, this amount was only for the actions inside this bioscience network. Maybe I'm wrong, but I think the total budget of the corporation, I'm looking at Perrine, is more or less 300,000 euro for the regional corporation, but in all the field, not only scientific, but also governance and cultural actions, etc. But here, no, this 30,000 was only for these actions inside the Rede of Bioscience.
And mainly goes to the travel scholarship, this money?
I don't know how it works exactly. No, I think it's so for the Brazilian event today it was for travel grant, but I think the envelope is for each country, each event, and then they organize their own events and budget. So it depends on the choice they will do with that program.
Over here. Yeah. First of all, you said, I understood well, this course now like 2 or 3 years that you are giving this course in Montevideo and so on. And first of all, what's the level of the students that usually are coming? The level, undergraduate, graduate, postgraduate course. And the second question, are you guys follow up them? Are you follow up the student to know how they are doing today, how they are using the knowledge that they learned? No? And the last one is this course stimulating collaboration between those students with you guys that are the seniors one, or between the research groups that they belong to? Okay, thank you.
Well, for the first question, our students are mainly postgraduate students. Most of them PhD students, also master students, but mainly PhD students. Also, we don't discourage people to apply independently of their career stage, but ideally to take the most profit of the course. You ideally need to be a postgraduate student. Also, there are postdocs that have come, or even early career scientists in some cases. But the main focus is on post-graduation students. And the other questions are very interesting because indeed, yes, we keep in touch with many of them after the course. And these courses have also generated, I don't really know if some interaction between the students, that is not, I don't have this mapped, but they have, this has generated collaborations with us, for example. Indeed, there is a particular case now, a girl that came to our course the past year, and then we keep in touch. She applied for a Calmet Yersing grant from Pasteur, and she got it, and now she's going to be spending 1 month in my lab working with metagenomic data and applying some of the things she learned during the course, analyzing her own data that is in the context of her PhD. So this is one case, but there are another ones. We don't do an active follow-up of every student, but for many of them, they come back to us to ask things and to to discuss results after they return to their places, and in some cases this has generated some ongoing collaborations. Yeah.
Yeah, thank you for the presentations. It's very interesting, your association. Do you think that the Pasteur Institute of French Guiana or Guadeloupe can benefit from your association? And how can this be done, sorry? And the other thing, what is the level for your course? Is it for the beginners or already confirmed bioinformaticians? And for confirmed, do you have something?
Well, indeed, I don't remember. I forgot to mention this, but we organized a course there in Guadeloupe that we sent Pablo, one of our researchers, together with Jan. That Jan has spent also some time with us in Montevideo, and I think was very nice. It was a metagenomic course for people in Guadeloupe, but as I mentioned before, the courses we plan in Montevideo, considering the demand we have, are for PhD students or post-graduation students that at least they have had some contact with bioinformatics, at least basic contact with bioinformatics. But in case we identify another kind of demand, for example, if there are people that want to start but they don't have any basis for bioinformatics, we can think about generating this content without problem and thinking about a different scope for a course. It's not a problem. But in general, for the courses we had in Montevideo, the students at least have some knowledge or some experience on Linux and on bioinformatics in general.
Concerning the interest of the Pasteur Institute of Guadeloupe and French Guiana in your association, do you think we can have exchange or something?
Sure.
I think that it would be possible. I think also that it is very important to think about the last question. Is it possible to go towards a bioscience network, AMSUD, with RIIP, to think it is possible to think in agreement of this association, focalized principally in formation of human resource. And we think that RIIP América it would be a very good point of access to the communication of all these actions.
I think it's a very good idea, and we need partnerships. As we said before, we in this region we can't stand on our own, and I think this is a very nice proposal. And making also the link with the courses that are being done, in the network and in other places, I think it's essential. Not to do twice the same, and to exchange teachers between the different networks. It's refreshing. It's getting new ideas, and then also being informed on what's going on in other networks, like the discussions we had at midday, to getting to know on networks that are working on subjects that we also are interested in this region. I think for the Pasteur International Network, it's essential.
Thanks.
And I think it's a subject that should be put on the table during the next directors' meeting.
Just for information, Kathleen, maybe you can say a word on that. The association each year spent 250,000 euros for the courses. Not only, I mean, the courses across the different regions of the world. And maybe you can give more information.
Yeah, well, for the ones who are not from the network and who don't know, yeah, it's one of the main activities of the Association Pasteur International. And we spend half of our budget on courses in the network. 50% of, I think that's also one of the questions that was asked before, 50% of the students are people from the network, and 50% is completely open for people from the country and from the region. And the label that has been presented before, it's you have to see it together. It's exactly to promote this kind of courses and to make easier link with universities and with existing university partnerships.
Thank you, Kathleen. Any other question? So I think we are done, and so the question will be addressed on the next General Assembly of the RIIP. Okay? Super. Thank you, everybody.
Well, the next step will be the opening, the official opening of the Pasteur USP platform, and there will be persons that will guide you to such place. We have some people like Patricia and some of the students that will organize, but we still have some time. We are advanced at half an hour or so, so you can just be prepared to move to another building that is very close to us. It's 300 meters from here, and there will be people helping you to go there. Thank you very much.